So welcome everyone to day 2 of the Autumn School in Neuroscience from Oxford University. It's the first time we've been online and we're really delighted to have so many of you with us, and my thanks to Nick Irving and Nicola Bridge for organising this in the background. Some of you may know that the Autumn School has a very long and rich history. It's probably 30 years old by my reckoning now. 1990 was when it first started. And many of us, many of you, were junior researchers or students when we first attended. And I'm delighted to say that we've had so many of you back as speakers, and I've ended up organising it myself. Anyway, we hope that you're going to enjoy day 2 along with our speakers, that you'll enjoy talks from areas you might not have engaged with before. And this, the sessions today are on evolutionary neurobiology and also on neuroanatomy, linking MRI to microscopy. And I'm delighted to have Robin Dunbar lead the session on evolutionary neurobiology. Many of you will know that Robin is Emeritus Professor of Evolutionary Psychology here at Oxford. He's a Fellow of Magdalen College and a Fellow of the British Academy. And he's put together a great team of speakers today to cover the topic of evolutionary neurobiology. So over to you, Robin. Thanks very much.
Okay, thank you very much, Masood. Welcome, everybody. This morning's session is really trying to take an evolutionary perspective on things to do with neurobiology. And I guess an evolutionary perspective here really invites us to step a little bit beyond just the mechanisms involved in the neurobiological processes that make us all tick, essentially to ask why those kinds of mechanisms exist and what functions they serve in terms of the individual's life history and ultimately their evolutionary fitness. An evolutionary perspective actually has the more than anything else, I think, the benefit of allowing us to pull together into one framework all the disparate, fractionated subdisciplines of psychology and allow them to integrate in a more helpful way that kind of poses questions for us that we perhaps wouldn't have thought of asking if we'd just focused entirely on the mechanisms, which is mostly what psychology does, perhaps. So I'm not going to say any more. Time is pressing for us today, so I'll start with the first speaker. Jerome Sallet has been a Royal Society Research Fellow in the Department of Psychology here at Oxford for a number of years now, and he's going to speak on some of the anatomo-functional properties of the social brain in humans and other primates. So, Jerome. Jerome, we've lost you.
Oh,
your picture anyway.
Here am I.
There you go. Right.
I'm trying to share my screen, which apparently I cannot. Yes, Nick can share the screen. All right, I apologize for that. Oh, here it is. Okay. All right. I hope you can see my screen now. All right, so thank you very much, Robin, for organizing this symposium and for having invited me. Thank you very much for joining this symposium online. I would have preferred to have you in front of me, but I will do it in front of my screen only. So today I will talk to you about the anatomo-functional properties of the social brains in primates, humans, and macaques, and I will more specifically focus on the temporal cortex, especially the cortex that lies within the superior temporal sulcus. So most primates are social species, and it is thought that the computational needs requires to live in large and complex social groups is one of the key factors that has driven the expansion of the neocortex in primates. All the processes that are specific to knowing and acting on information about social companion are thought to rely on a set of brain regions that are often called the social brains. And those regions, like a subset of them, are represented on this schematic view of the human brain. So these regions are located here in the temporal pole. They extend into on the bank of the superior temporal sulcus, extending here towards the temporoparietal junction, also located in the prefrontal cortex, here on this medial view on the medial prefrontal cortex. Involves also some key subcortical structures, such as the amygdala. So, and among all these social processes that have been investigated, maybe one of them has attracted a lot of attention over the last 40 years. This one is the mentalizing, the theory of mind ability, so our ability to represent others' thoughts and believe others' cognitive states. Here, the Sallet and Anne task, which is a classic task that has been used to investigate such ability in human subjects, and requires on telling subjects some stories about, for instance, here, Sallet and Anne. And Sallet has a marble, she puts the marble in the basket, and then she goes away. While she's away, Anne takes the marbles away from the basket and hide it in the box. Then Sallet comes back to the crime scene, and the experimenter is asking the subject to guess where will Sallet look for a marble. If you think about what Sallet knows about the state of the world, you will answer that she will probably look into the basket where she last saw and when last put the marble. So if you want to investigate this ability from an evolutionary perspective, you will have to come up with a version of this task so you can test it in non-human primate, for instance. So you can first start by reading the story to your monkey, and at best, you will end up with a laughing monkey. But if you come up with a language-free version of the task, your subject might be able to perform the task, but you will end up with a grumpy colleague that will complain about what you just achieved, and that will claim that you didn't manage to test mentalizing ability in the primate. And so, stepping aside from designing a new task, what we decided to do is, rather than looking at the function, maybe we could first start looking at the structure. Is there a specific structure, a set circuit that is supporting this function in humans? And if we can identify such circuit, could we identify the similar circuit in a non-human primate brain? So in humans, we know that there are at least two key regions that have often been associated with mentalizing abilities. There's this region here located on the caudal part of the lateral surface of the brain here. This is the temporoparietal junctions represented on this lateral surface of the human brains. And there are also the medial prefrontal cortex. So what we want to know are the specific architecture associated with these two regions and whether such a similar architecture could be identified in other species. Okay, so here we are following the lead, following the footsteps of Dick Passingham and Rolf Skirter-Wells. What they did in their work is they established that each cytoarchitectonic area has a unique set of input and outputs, and that unique set, called the cognitivity fingerprint, is crucial for constraining the functions an area can perform. So, for instance, here you've got two cognitivity fingerprints of area 14 and area 9, two areas of the prefrontal cortex are represented on this spider plot. Each arm of this plot is a brain region, so area 10, 11, 12, 13, etc. And the length of the red bar on this spider plot corresponds to the strength of the connection between area 14 and the given area. So here, 10, 11. As you can see, the spider plot, these cognitivity fingerprints for the two areas are completely different. Area 14, an orbital frontal cortex region, is connected with other orbital frontal cortex regions, while has little to no connection with area located on the lateral surface of the prefrontal cortex. However, area 9 is part of the lateral prefrontal cortex and has connection with those areas on the lateral surface and weaker connection with area on the orbital frontal cortex. Okay, so initially, this has been designed so to investigate the function of different brain regions within a given brain, for instance, a human brain. But we expanded this approach, and now what we are doing, is we are using this signature of a given area from a given species, and we are looking for, in another species, whether this signature could be identified, whether this fingerprint could be identified. We are doing it by by conducting resting state fMRI analysis. So we extract the time course of the bold signal within a given brain region, like called the seed region. Seed region. And we are looking where else in the brain could we find voxel, our unit of analysis, that share this similar pattern of activity. The result of such analysis is a functional cognitivity map that represents the regions that are functionally coupled with these seed regions. Out of it, we could extract the strengths of the coupling, like a Z-score, and we can plot it and compute this cognitivity fingerprint for various target regions. We can do that, looks like, okay. We can do that in humans, but we could also do that in macaques, for which we have also collected resting state fMRI analysis. And because we know that some regions are homolog between the two species, what we can do is we can compute a cognitivity fingerprint for humans and macaques in the same regions, and then quantify the similarities and difference between the two species. Okay. So we first did that for the medial prefrontal cortex, one of the regions that is important for mentalizing in humans. Hope you can see it. So this, on the left, you've got the cognitivity fingerprint for the human area 10. So the hotter the color, the stronger the coupling. Area 10 is coupled with other region of the medial prefrontal cortex, also with a region of the dorsolateral prefrontal cortex on this lateral surface of the human brains, and with anterior region and the region of the anterior part of the temporal lobe. In the macaque, you can find some similar cognitivity pattern, with coupling with other region of the medial prefrontal cortex, region of the lateral prefrontal cortex, and anterior part of the temporal cortex. We could compute the cognitive fingerprint using homolog regions. You can see here I overlaid the two cognitivity fingerprints. You can notice that there are similarities, but also some differences. But if you look throughout the macaque brains, and if you compute the degree of dissimilarities between the cognitive fingerprint of all the areas of the macaque prefrontal cortex with the human area 10, you will find that the area that share the most similarities with human area 10 is the macaque area 10. Okay. I would be happy to talk to you more about the medial prefrontal cortex if you want. So if you're looking for questions to ask me at the end of the talk, here's one, ask me about the paracingulate sulcus. But for the sake of this talk, I will move on to the temporoparietal junction, which is the other key area that we investigated. So the temporoparietal junction is an heterogeneous region. It's not an homogeneous region, and it can be subdivided into 3 sub-areas. This was done initially by Jorge Mas using diffusion-weighted imaging data. And this study, this was replicated recently by Matthias Schultz, was a postdoc with us and is now a professor at Innsbruck University. So he was able to identify the 3 sub-area that the TPJ is made of, or this region is hosting, well, if I could say that. But in addition to just doing this resting state fMRI parcellations, he was also able to collect some task fMRI data in a subject. So he used several localizer, some tone localizer, like a false belief localizer, where you are telling stories about people to subjects, and at the end, you're asking them true or false question, so about this story they just read. Or you could use non-social story that does not involve people as a control. You also use the social animation task, in which you have geometric shapes that are interacting on the screen. And the way, for instance, you would describe this video will be that the red triangle is pushing the green triangles away from the square. So you will attribute some intention to geometric shape. Or you have those shapes that will be moving randomly on the screen, bouncing around the screen against the wall of the square or the edge of the screen. There's some non-Tom tasks, the biological motion, so just dots that are moving around in a kind of like a biologically friendly way, or scrambled dot motions, and then a default mode network localizer. So the default mode network is a set of brain regions that have often been associated or linked to the social brains, and there are regions that are more active when the subject is at rest compared to when he is performing a cognitive task, for instance here, a working memory task. So when you look at what's happening in this big temporal parietal junction mask for this different task, first of all, if you look at the false belief localizer, you can see the hotspot of activity is in fact mostly located on this false belief localizer is mostly recruited voxels that are located in this posterior subdivision of the temporal parietal junctions. These voxels also have been also associated with the default mode network localizer. However, the other two localizer are recruited distinct, having distinct pattern of activity and recruiting distinct voxel. So here the take-home message is like, if you are investigating this mentalizing ability in humans, you will most likely recruit voxel that are located in this temporal parietal, posterior temporal parietal cluster, a cluster that is associated with the social brain, so it has strong connection with the posterior cingulate cortex and the medial prefrontal cortex. Okay, so now we have a signature for our temporal parietal junction, and we have been also able to assign a function to this area. So now we can search in the macaque brain whether we could identify an area, brain regions that share a similar connectivity patterns. So in this study we conducted with Ramos a few years ago, we used in this first analysis, we used very simple cognitivity patterns. We knew that this temporal parietal junction area, the posterior part, is well coupled with the posterior cingulate cortex, as I just said, and the anterior cingulate cortex. But it's not talking at all with this mid-cingulate cortex or the anterior insula. Those four regions are they have their homologues in the macaque brain, so we can search for we can first search for where are the regions that share this type of connectivity pattern in the human brain, and we have identified this temporal parietal junction in an independent set of subjects. And if you look at whether there are regions with that pattern in the macaque brain, you will identify voxels that are located in a slightly odd way. They're not at the back of the superior temporal sulcus, they're located in the mid part of the superior temporal sulcus. Okay. So I didn't go into too much details, but with Matthew Rushworth and Jorge Mas, we have shown that the medial prefrontal cortex in macaque and human do share common functional cognitivity property. So there's no new area in the human medial prefrontal cortex that would explain to support this mentalizing ability in humans. If you're investigating, if you're interested in the temporal parietal junction, you should be careful. This area is heterogeneous, and you should subdivide it and be accurate when you're talking about which subdivision you are talking about. And it's just the posterior part of the temporal parietal junction that is associated with mentalizing process, with high order social cognitive process. This posterior TPJ in humans, you could as not an homologue, but as an area that share a lot of similarities in the macaque brains. This TPJ-like area is located in the mid part of the STS. But based on those works, this neuroanatomical work, we can simply say that there are similarities between circuits supporting social cognitions in macaque and humans, suggesting that the macaque brain contains a precursor of the human social brains. But we cannot claim that a macaque does or does not have theory of mind, because we didn't investigate the function. And that's what we aim to do in the next set of studies. First of all, this study that is almost 10 years old, in which we had the opportunity to test whether changes in the social environment could cause some plasticity, could cause some changes in structure and function of the brain circuits in the macaque brains. So we had animals that arrived in large social groups from the breeding facility, arrived to our research colony, and after they settled to the Oxford life, they were reassigned to different research groups and research projects. And again, after at least 3 weeks in the newly formed social groups, some animals were scanned for different research projects than this one. But we were able to use this data to look for potential correlations between size of the social network the animals are living in and gray matter volumes. And we found some positive correlation in several parts of the brain, in the temporal cortex and as well in the prefrontal cortex. The region that were in which the voxel in which we found this correlation between size of the social network and gray matter volumes are represented on this set of coronal section of macaque brains. They are located here in the superior temporal sulcus, in the region we identify as the TPG-like area, also found here in the dorsolateral prefrontal cortex, in the major prefrontal cortex, and in the rostral prefrontal cortex. And here is a representation of the type of correlation we observe between the size of the social network and our proxy for gray matter volume called the determinant of the Jacobian. Not only did we observe some changes in various parts of the brain, but we also noticed that there were changes in the way those brain regions were interacting. So if you look at the resting state data and you place it in the superior temporal sulcus, we'll find that these regions is coupled with the medial prefrontal cortex and with the anterior cingulate regions. But the way those regions are interacting is modulated by the size of the social network. And so the larger the social network, the stronger is the coupling between those two regions. And this is represented in this bottom right plot. Okay, so the mid-STS seems to be important for social cognitions. Could we go into more details? Could we find out more about it? So the next approach we used is a neuropsychological approach. So what we try to do is to perturbate, or you try to remote on the social behavior. We first use this task that has been developed by Pete Rudebeck and Matthew Brushworth. So it's a very simple task. The monkey is in a transport box. In front of him, there is a transparent perspex box on top of which you placed a food item. Okay. And in the box, you place an object, that could be a neutral object, or you can display a video. The idea is that the more valuable the object or the video is, the longer it will take for the animal to reach the piece of food, Here we looked for we looked at social videos versus neutral videos or neutral objects. You can see on the right, you can see that the control subjects are faster to reach for the piece of food if you're displaying a neutral video compared to a social videos. The same is observed after mid-STS lesions or after an OFC lesion. However, after lesion of the cingulate gyrus, this effect is abolished. More recently, work from Betsy Murray lab has shown that this is the anterior part of the cingulate gyrus that is critical for this social valuation effect. So here we are able to dissociate the contribution of the mid-STS to the ACC, but we still don't know what the mid-STS is important for. So we asked our subject to perform a second task, in which is a two-choice task, in which they could choose between the blue target that will reward themselves or the red target that will reward themselves as well. But a second reward will then be given to the monkey partner that is in the transport box next to him. So monkeys like control, we test 3 groups of monkey, control monkeys, monkey with an OFC lesion or STS lesion, mid-STS lesion, sorry, I should say. And they perform at different levels. But what's important is what happens when you replace the monkey partner by a container. So it's a non-social partner. Then the control, they reduce drastically the amount of time they pick the red option, the pro-social options. Same occurs in the OFC lesion animals. However, the STS lesion animals, they do not really adjust their behavior. So the mid-STS seems to be important for adapting behavior to different social contexts. Okay. Last one is we're going back to our questions of do monkey represents, could monkey mentalize? And how do we test that? So recently there are two groups of scientists that came up with a monkey-friendly version of the Sallet and task. But as expected, they were able to find that some of the animals, not all of them, but some of the animals were able to pass the test. So the way they did it is they didn't tell the story, but they presented movies to the animals. And here, instead of asking a question of where Sally will look for the marble, they look at where are the animals looking for. And the first gaze, especially, are they gazing towards the box, toward the box where the item is, or towards the basket where Sally is likely to look for the marble. Okay, they have been criticized, so we didn't follow that route. And we went to find some inspiration from computational neuroscientists, especially from the work of Rebecca Sachs, who thought that mentalizing ability and what's happening in the TPJ should be or could be interpreted within a predictive coding framework. And this is an example from one of her experiments in which they ask subject to read stories. The first part, like they're setting up the stage here. You read the story about you grind up some peanuts, you add that to the dish, you serve everyone, but one of your guests is allergic to peanuts. And then they scanned, they especially look at the activity in the brain when the subject read the second part of the story. Either you knew about the cousin allergy, so you did it on purpose, or it was an accident. And this intentional versus accidental situation is associated with a differential change of response in the TPG. Closer to Oxford, like Tim Burns and Matthew Rushworth, they asked their subject to perform some social gambling task. And when they compute a social prediction error, again, they were able to find some correlates of this social prediction error here in the TPG. So what we did with Léa Romazet, who was a PhD student, we tried to see if we could identify a type of social error signal in this mid-STS region. What we did, the very crude social prediction error, because we used a free viewing experiment in which we presented social video to our animals, but we controlled the content of the social video. So a social video, what you could predict the end of the social video from its first few seconds. So if you see animals grooming, you would expect that a few seconds later, they might still be grooming, or at least resting next to each other. So that would be a predicted end of a social video. However, if you start seeing animals grooming, and then suddenly one is biting the other one, that will be quite unpredictable. And so in this block design experiment, what we did is we contrasted the activity recorded during this viewing of unexpected social video with unexpected ended versus social video with expected ending. And what we were able to record is differential activity here in the superior temporal sulcus. However, this might be simply driven by low visual features like motion. So we run additional control video in which we presented the same unexpected video, but this time just a scrambled version of this. And then we had a cluster of activity here in the posterior part of the STS. But we still have this anterior cluster that seems to be specific to the unexpected social situation and not explained by a low level visual feature. We then presented to the animals another control to test whether this activity recorded here is specific to the social situation. So here we presented to the animals non-social situations, so object interacting in an unexpected or expected fashions. And then at a slightly lower threshold, we're able to get a cluster of activity here in this region that is called the MT complex, a region that is sensitive to motions. On this bottom figure here, we have the overlap of all our effect. So in green, this activity listed by this object contrast, in yellow, the object contrast, in green by the scramble videos, and in red by the social prediction videos. And you can see this anterior cluster that are only recruited when you play the social prediction videos. Importantly, some of the cluster, some of the effects are located where in this part of the mid-STS that we identify as the TPG-like area, and that is represented by this white circle. Okay. So this was conducted on 14 animals, a large group of monkey, and Leah did also performed subject by subject analysis. And then on this figure, what is plotted is the superior temporal sulcus, the like unfolded temporal sulcus. Here you have the left temporal sulcus and the right temporal sulcus. And what you have is this social prediction, clusters that are in fact located on the dorsal bank of the superior temporal sulcus towards the fundus. And what is also important on this figure is that she represented the peak of the face patches. And what we have here is that face patches like brain regions that are more specifically recruited when you present faces to subjects, the well-known feature of the temporal cortex in primates. But what we have is our social prediction cluster that are distinct from these face patches. And I will come back to that in a second. Then together with Leah, and as well as with Matthew Loskevich, who was a master student, and with Halim Mahmoudi, who just joined Oxford, we conducted an additional control experiment in which we decided to perturbate the activity in different brain regions that we thought could contribute to explain the effect we observed in the mid-STS. And for that, we used focused ultrasound neurostimulation. So if you've got 1 minute, I will briefly explain how it works. So it's a minimally invasive method that we developed with Jean-François Aubry and together with Davide Foloni and Matthew Rushford's group in Oxford. So this, you place the transducer on the scalp, and then you will send ultrasound wave through the skulls, and those ultrasound wave will mechanically impact on the neural tissue, perturbing the normal activity, and this perturbation could last for up to 2 hours. Importantly, this method allows you to target deep brain structures such as the amygdala, and here's the simulation of the effect of the transcranial ultrasound stimulation of the amygdala we did with Davide Foloni. So we did a bilateral stimulation, and what is represented here is the functional cognitivity map for our sham stimulation on the top. And after our amygdala stimulation, as you can see, there are some big changes here, like increased anticoagulation with the MCC after you stimulate the amygdala, or here you have a radius coupling with the ventrolateral prefrontal cortex after stimulation of the amygdala. Here are the cognitivity fingerprint of this effect in yellow after the stimulation of the amygdala, in blue after our sham sessions, and in red, that was the control side. Okay, so we know that when we stimulate brain regions, we could impact on the circuit. It changed the coupling this region as with other brain regions. So here we thought that because we had some effect as well in the frontal eye folds, that maybe the mid-STS effect we noticed might be related to an additional effect. And we also that's one of the main questions we wanted to know. We know that the perigeneal ACC and the STS are also interacting with each other. And so we wanted to stimulate that region as well. And the amygdala is a part of the social brain. So what we aim is we stimulated our animals in these 4 different conditions, sham, or stimulated one of the 3 areas, and then we presented them with the movies. And so what we noticed first, I hope just by glancing at this picture, you can see that there is not much difference between the different conditions, although they're especially in the mid-STS, like there is no difference. There are differences, so our stimulation worked. There are differences after stimulation of the amygdala, of the peritoneal SCC, for instance, you see some differential effect in the anterior STS, the hippocampus, but also the frontal pole. The stimulation of the FEF, there are also some changes in response of the frontal cortex. But again, there is no significant difference in the mid-STS. So the mid-STS, I think it's unlikely to be explained by an attentional effect. When you disrupt the activity in this attentional system, you don't disrupt the activity, you don't change the response in the mid-STS. Okay, just to go back to the faces. So the face patch is a well-known characteristic of the temporal pole. They are located along the bank of the supertemporal sulcus in the macaque. In the human, they are located in a slightly different position, but they are also in the temporal cortex. But one of the key difference between the macaque and the human is, like, if you look at the cognitivity, functional cognitivity map of these regions that are sensitive to social prediction. And so this mid-STS, or we call slightly anterior STS, so we place the seed, that's the black dot. You get coupling with the medial prefrontal cortex, the PCC, but also with regions of the occipital area, occipital cortex. And this is not at all what you observed if you look at the functional cognitivity map of the TPJ in the humans. There's difference here. As we said, and as we showed, this region, the region sensitive to social predictions and the face patches are distinct in the human. So what you can do is you can conduct a partial correlation analysis, trying to identify what is the connection that is specific to the superior prediction area, and what is the connection that the connectivity that is specific to the face patches. They are located together, so they are likely to talk to each other, but maybe they have what you see here is the fact that those two systems are talking to each other. And if you do a partial correlation, then you will be able to tease these two systems apart with this area that are sensitive to social prediction, that are more like default mode networks circuits, and with the face patch area that are more like a visual area talking to the occipital cortex. So what we might have is a system that has, since the last common ancestor to macaque and monkey, becoming less dependent on maybe visual input, like less talking to the visual system. Oh, okay, and among other things, that's just wanted to briefly point to this work done recently by Lea Rumazai and Jorge Mas about the evolution of white matter pathway in the temporal cortex. So it's not just one thing that has happened to the temporal cortex, there are several things, including some specialization within the ILF between the lateral and the medial sub-pathways since the last common ancestor to macaque and humans, and of course, a huge change in the arcuate sulcus, probably related to the emergence of linguistic ability in hominids. If you want more information about it, I just go to their very nice paper. Okay, so to conclude, so we showed that the mid-STS and the anterior cingulate cortex support distinct social, are associated with distinct social functions. The anterior cingulate cortex is important for evaluating social information, while the mid-STS seems to be more important for adapting behavior to different social contexts. If you interpret the activity of the TPJ within this predictive coding framework, then we have been able to identify that the mid-STS is coding for a social prediction error and not an attentional effect, as when you stimulate the FEF, you're not altering the response of the mid-STS. So it suggests that maybe there is, jumping to the conclusion already, but it suggests that there is a precursor of the neuronal architecture supporting complex social cognitive process in the human brain. So it's this precursor of this neural architecture is already existing in the macaque brains, and it seems to have the same connectivity, but also support similar computations. Of course, the temporal frontal pathway have expanded since and changed since the last common ancestor to macaque. So there's more work to be done to fully understand the evolution of this system. All right, thank you very much. I would like to thank all my collaborators, and I would like to thank you for your attention.
Thank you very much, Jerome. If you'd like to unshare your screen. We've got a minute or two for questions here. If I can just find them again. Okay, so I'll crikey, they're coming in so fast that the whole thing keeps moving. All right, I'll just take one at random here. Actually, this is from Prenatal. What are the limitations or difficulties faced by the study of evolutionary and comparative approaches, and presumably studying these effects? Would you like to comment on that?
Difficulty. One of the difficulties is that we don't have access to some of the extinct species. So what we are doing is comparative anatomy, and we are inferring about evolution. That's one of the difficulty. And the other one, when we are trying to test different species in different tasks, like are we using the right task, and in terms of design, are we asking the right questions, and is the task adapted to the ecology of the animal? That will be the main.
Okay. All right. I'll just slip one more quick one in. Come on.
Oh, and then if I can just add something.
Okay.
In humans, you read the instructions so they can quickly do the task. In animal models, you have to train them to perform the task. You have to guide them, so it takes ages. So are we exactly? Yeah, that would be.
Yeah. There's nothing harder than working with animals on anything. Okay, just one quick one from Prashant Famuri. Do you think that second order consciousness is possible in macaques? And I interpret that as meaning self-consciousness.
That's a really
in the sense of being able to reflect on. It's theory of mind, really, isn't it? Being able to reflect on your.
Yeah, can I ask you to answer that question for me?
No. I was hoping
that's a really good one. Again, they don't pass the mirror task unless you train them a lot. I think Moving Poo has some interesting data about it. I think Franz Deval mentioned that if you are self-conscious, you should do like you would use maybe a mirror to inspect body parts that you cannot see otherwise. And we had one monkey, but just one out of the many monkeys I work with, that used a mirror for dentistry. She was inspecting a gum.
Right.
So
yeah. Okay. Yeah.
Less than human, for sure.
Yeah. But I'm not convinced by the mirror experiments. I think they just tell you whether they understand basic physics.
Yeah. But then if they inspect like that, that comes like, well, of course they're using it to see how it is happening from behind. It's like if you go into a research facility, all the monkeys are really good to check who is coming using the mirror. But
yes,
self-consciousness. Yeah.
Okay, I think we should move on to Sophie Scott.
Thank you,
Sophie. Are you ready to unmute yourself and bring yourself up? So Sophie is Professor of Cognitive Neuroscience at UCL in London. She has the very dubious distinction of having challenged me once to do stand-up comedy, which I duly rose to the challenge. I got the t-shirt, but I'm never going to repeat again. And she's going to talk about the neurobiology of speech and laughter.
Okay. Can you hear me, Robin? Can you hear me?
I can, yes.
Right. I'm being faced with what everybody hopes they'll see when they're doing a Zoom talk call, which is my computer asking me for my password so it can do something. So let's just see if that works. Okay, I'm going to start my talk. We may or we may not have sound. Let's see how we get on. So I'm going to talk about the neurobiology of speech and laughter from an evolutionary perspective. And this is going to be a fairly general story, and it's going to capture a lot of the stuff that I'm kind of interested in in my lab. So if you have any questions going into more detail about any of this, please feel free to ask either now or drop me an email. And I'm interested in speech and laughter because basically I'm interested in voices, and actually voices are a very highly conserved aspect of mammal evolution. So what you find is that the larynx, sorry, I can hear Jesus Christ Superstar being played in the background. I'm just going to ask my son, who's also at home, to turn this music off. Hector, turn the music off, please. Thank you. So, because I'm giving a talk, Hector. Right, sorry about this. So what I'm doing here now is talking, and I'm using my larynx to produce the source of the sound. And in fact, that is a highly conserved mechanism for producing speech and voices and making sounds across mammals. So the larynx has evolved to be a structure to stop things from falling down into the lungs and from blocking the lungs off so you can't breathe. And what almost all land mammals have found is a way of sort of reversing that procedure to close the larynx, blow air through it, vibrate the larynx and make a sound. And that gives us this very general model for the mammalian voice, which is called the source filter theory of vocalizations. So you have the larynx, which is closed to make a sound, or partially closed, and then air is blown through that. That gives you the source of the sound, and then you shape that sound with everything above that. So that's things like the shape of the mouth or the angle of the jaw, and that's altering the source sound that you make down at the larynx. So if we look here, here we've got a red deer. Red deers, like humans, have got larynxes that are movable and can be taken down very low. If we look here at a video, so you may just be able to make out there. First of all, Robin, can you just tell me, could you hear that? Was that making a sound for you? Because otherwise I need to know if this is not making a sound. Can anybody tell me if this is making a sound?
It's making no sound.
Okay, that is very useful for me to know. That's a deer making a roaring sound. And if I just play it again, bearing in mind I know you can't hear it, what you can see...
Now we can hear it.
Excellent, thank you. You can see the larynx moving up and down, and that's the larynx being able to change aspects of the pitch of the sound that the deer is making. But you can see the deer is just kind of throwing his head back, and he's not doing much else above that. So that's giving you a classic example of a mammalian vocalization. Now, what's happened in human evolution is one of the things that's had a very big impact on our use of the voice in this way is the fact that we walk upright. At some point, bipedalism became a factor of human behavior, and this has affected a lot of the different sort of anatomical aspects of humans, but also actually of our brains. And you can see this quite strikingly in terms of our voices. So one of the big reasons that bipedalism affects human voices is that it frees up our rib cages, and our rib cage is very, very important to our voices. So what you find in the rib cage is there are muscles between the ribs, the intercostal muscles, the inner and the outer intercostal muscles, work in quite a complex way to pull the rib cage out and up, creating negative pressure inside the lungs, which pulls air into the lungs. You then relax the muscles back down and air is pushed back out. So this is a structure for breathing in mammals. This is how breath is got in and out of the rib cage. This is how we stay alive. You're all doing this right now. It's called metabolic breathing. Don't stop it. And if I was to put a breath belt on you so I could actually look at the movement of your rib cage while you breathe in this metabolic way, you get this very smooth movement. And what you find in humans is because we walk upright, unlike all other mammals that to some extent or another have to engage their forelimbs when they're walking, certainly for land mammals, but walking or sort of staying upright, humans don't need to do this. This has freed up our forelimbs, so we have hands, which are fantastic tools, but it also has freed up our rib cage from having to support our weight. So humans have a control over the intercostal muscles that has been made possible by the fact that we do not need to use these intercostal muscles to support our weight. And when we look at humans speaking or using their voice in any way, one of the first things that you notice is we have a completely different pattern of breath control that underlies this. So if I now take this same measure, looking at the movement of the rib cage by just looking at this overall movement of the expansion and contraction of the rib cage, I see a very different pattern when I look at someone speaking. If I speak and take a breath in, now, instead of what we see for metabolic breathing, what we see for talking involves a very different movement of the intercostal muscles. We take a breath in and then we use those same intercostal muscles to prevent air from rushing out through the larynx so that we can make a sound when we speak. And if I keep talking without taking another breath, you can hear I've stopped, work really, really hard to make a sound. Now, that's what I'm showing you there. That's me talking and talking and talking until I run out of air and then taking a very big breath. And it's very important to realize what exactly this is doing. What this is doing is it is allowing humans to maintain what's called a constant subglottal pressure. So we are able to make a constant sound at the larynx by how we're breathing out in this controlled way. And it also gives us a much longer window over which to do this. And you may not realize this, but when you're speaking or when you're singing, the melody, the pitch and the rhythm of your voice is all coming in actually from this pattern of breath control. So it's giving us melody and rhythm in our voices, and it's also giving us these longer periods over which we can use our voice. So this is critical. If we couldn't do this, we would have to breathe. So even if we could speak, we would be speaking on every single exhalation. And there are examples of animals. There was an elephant in Malaysia a few years ago who had learnt to copy human speech by putting his his trunk inside his mouth and rattling air, but he would produce one word at a time because he was an elephant and he didn't have this extended breath control. I don't know what all the fuss was about. So he doesn't feel like it, but in fact the first and most important thing that's happened in human evolution that let us have human voices, and really there isn't much in nature quite like the human voice, is in fact that we walk upright and we have able to have this breath control. And of course this breath control isn't just to do with the anatomy and the fact that we walk upright and we freed up our rib cage from supporting our weight. You have to have the neural control as well. And you can actually pick up in human evolution when we got the breath control that allowed us to start using our voices in this way, from looking at an expansion in the rib cage that's associated with a greater brain control over this area. You get the same thing for control of the fingers and the hands. And in fact, you have as much fine control over the intercostal muscles as you do over your fingers. That's how precise these movements are. There are other things that have affected human voices that have happened in our evolutionary trajectory. So if you look at the faces of primates compared to the faces of other mammals, they have much flatter faces and much shorter mouths. But even compared to other primates, humans have got very, very flat faces. So this is a human compared to a chimpanzee. And if you look at the shape of the front of the face, you can see how flat the human face is. This has one very big consequence, and it sounds like a trivial consequence, but it means we don't have to have very long tongues to get to the front of our mouths. Human tongues can be short. And this is important because when we speak and we sing and we do things with our voices, we very commonly use our tongues in very complex ways. So our tongues are short, they're actually very flexible, and they're flexible for a couple of other reasons. So if you feel the roof of your mouth, you'll likely feel a kind of domed shape. If you look at the roof of the mouth of any other mammal, it is flat. And that's because the job of the roof of the mouth is so that you can eat food without things going up into your nose. The domed roof of the mouth means that the human sort of mouth space, buccal cavity, is short and it is deep. And this is giving us this kind of domed space in which we can move our tongues around and shape that source sounds that we produce in a much more complex way. And the other reason we can do this is that humans have a permanently descended larynx. So the larynx of almost all other mammals is sitting up right at the back of the tongue, anchoring the tongue at the back of the throat. And that's not the case in humans. This is why humans can choke. We have this descended larynx. It descends quite early in our lives. And this gives us a pharynx space back here. Other mammals don't have that. And it gives us a much more flexible tongue because the tongue is not anchored in this way, but it also means that we can choke. It means a couple of other things. It means we have a longer gap between the larynx where we're making the source of sounds and our lips. So we have a longer pipe to make the sounds of speech or anything else with our voice. And of course, in adult men, in adolescence, men go through a secondary descent of the larynx. So the male adult voice is deeper and has a wider spectral range than adult females. It's in fact the biggest source of dimorphism between male and female bodies in adulthood for humans actually is in the voice in this way. So we've got these other things that are giving us a very different kind of machine for making sounds, and that gives us, in fact, a very different thing we do when we speak compared to other animals. So this is a human talking in a scanner. We're just running the scanner like a video camera so that you can see the inside of her mouth as she's speaking, and just look at what the tongue's doing. It is continuously deforming. It's like an octopus tentacle. It's a muscular hydrostat. And all other mammals have got big, thick tongues that have to get to the front of the mouth. They cannot make these movements. So we've got this instrument for making sound, and it's working in coordination with all the other articulators. So the jaw, the lips, the soft palate there, and the larynx are working in this very highly coordinated way, along with the breath control, to give us this extremely complex signal. So this is what I mean when I say the human speech is the most complex sound in nature. I think it's probably more fair to say human voices are one of the most complex sounds in nature. And you can see it's very tightly linked to these different evolutionary adaptations. Critically, everything that we use when we're using our voice in this way, we already had. You can see now we haven't evolved something else to develop our voices in this way. It's all built on things that were already existing in other primates and other mammals. Now, for comparison, I want to compare this to what happens when we produce a different kind of vocalization, because I'm emphasizing here human speech, which is, as I say, kind of unparalleled in nature for its complexity. If we look at a different kind of vocalization, we see a very different pattern. Now, this is an example of a non-verbal emotional vocalization. And non-verbal emotional vocalizations are the sorts of things we do when we are in very extreme emotional circumstances, like you're screaming with terror, something disgusting happens and you go ugh reflexively, or in this example, when you laugh. So these non-verbal emotional vocalizations are more like animal calls than they are like speech, because they are produced in the same way. Look now at the articulators. The mouth is open, just like that deer's mouth. But now nothing else is happening. The tongue is just sitting at the bottom of the mouth. You can see quite a lot of movement going on, because you do move a lot when you're laughing, but it's not associated with any of these fine, complex pattern of control associated with what's happening when we normally speak. Now, this sounds lovely because it's laughter, but something like this would be there for most different kinds of non-verbal emotional vocalizations. They just don't normally go on for quite as long. So, of course, you would expect laughter to be like a non-verbal emotion, well, to be like an animal call, because we're not the only animals that laugh. It is rooted in an older evolutionary history. I'm not going to go into this in too much detail, but we see laughter emerging, for example, in human babies when they're tickled, when someone plays peekaboo with them. But certainly for tickling, that's also very much the case for many other animals. So it's something that's widely observed when laughter is first seen in chimpanzees. It's associated with tickling, and that's even the case for rats. So it has this older evolutionary history than us. It's a sound or a behaviour that is produced by other animals, other mammals certainly, often associated with things like play. Oh, this is just a very quick clip. This is just showing you what primate laughter looks like. So this is the late comedian Robin Williams meeting the signing gorilla Coco, who in a really humorless way asks him to tickle her. The other thing you'll notice is she's really interested in human nipples, and she keeps kind of delving into his t-shirt. It's hard to tickle a gorilla. She's so bored, she's looking at his t-shirt for a while here. But when you manage to get the gorilla actually finding the ticklish, start seeing how familiar that behaviour is. So there you go, look at those, her face, her movements start to look extremely like human laughter. And what have you got going on here? Let's not waste each other's time, shall we? So as I say, laughter is older than us. You find it in other animals. It's strongly associated with play. Play is a very, very common mammal behavior in juvenile period, but also some mammals play their whole lives. And it's so important to show that you're playing, to disambiguate behavior. You get things like play face, these very loose, open, happy smiles that you're seeing on the dogs. You also get, this is a play bow. When dogs do this, it means everything after this is going to be a game. I might growl, but I'm not angry with you because we're having a game. And it seems to be something that's a very important, when there's a sound associated with this, it's the sound of laughter. So Panksepp, who did the really nice work with the rats, pointed out that laughter sort of has, where you find it across mammals, always seems to have this role of, at its heart, of at least an invitation to play. And it's very interesting if you go back incidentally and read Charles Darwin on human emotions and emotion in other animals, because he talked a lot about laughter as well. And he thought that it was an expression of joy. And there is something joyful and playful about where you find it. I'm not going to go into this in too much more detail, but I think it's interesting to think more about what I say. It's an emotional expression. What's it an expression of? Well, Darwin thought it was joy. Panksepp thinks it's play. Those already seem quite closely linked. And I'm going to give you another big clue in a second. So this is just showing you, the risk of boring you silly, how different laughter is from speech. So laughter here is just an example of an emotional vocalization. We're seeing very different patterns of breath control. So that's talking, as I showed you before. This is laughter. You get this kind of zigzag, zigzag, because you're just squeezing air out from the ribs when you laugh, that you just get these very big contractions of the rib cage, and nothing else much happens. As you could see in that MRI, there's no, if you look at a spectrogram, you get a lot of detail being introduced into the speech, because the filter in the source filter model is very complex in speech. It's changing all the time, and it's a very simple one in laughter. So just to show you that pattern again, this is the pattern of breath control now. That was me talking, and that's me starting to laugh, and that's just showing you in more detail. Each of the ha, ha, ha sounds in laughter just come from single large contractions of the rib cage. It's a very basic way of making a sound, consistent with the fact that it's older than us, and it's a very basic mammal vocalization, most likely. Now, if we think about what this means in terms of brain control, we know, as I said before, we have a different kind of pattern of neural control over our rib cage compared to other animals that are not bipedal, other mammals that are not bipedal. If we look at the voice in more detail, you can see more of this going on. So this is the control network for vocalizations in other mammals. And moving beyond breath control, what you find in other mammals is they make sounds. They tend to make highly conserved sounds, so they don't tend to vary or learn from experience. They're often sort of, what, for want of a better phrase, kind of innately pre-programmed sounds, not always, but often. And they're associated with a brain network that runs in a midline system. So it's involving the periaqueductal gray, it's involving the anterior cingulate, and that's a network that is involved in this use of sound in other mammals, which is often, not always, but often highly reactive, frequently emotional, a bit like our emotional vocalizations, not necessarily under any kind of intentional control. In contrast, when humans speak or sing or rap or beatbox or do something else with their voice that feels more specifically human, the kind of thing we talk about when we talk about voices, that's associated with humans in a very, very distinct network now that sits in lateral areas of the brain, on the left and on the right. It involves sensory motor cortex on the left and on the right, because in terms of the sort of this very different control, not just of breathing, but in fact of the articulators, you get supplementary motor cortex, you get auditory cortex, because there's some auditory and sensory guidance of the sounds you're making. And this is strongly dedicated to, well, most obviously speech, not only speech. But if, for example, you lesion elements of this network, you can have a problem in speech production. It's associated with what's called an expressive aphasia. And in those situations, that midline context, that midline system for reactive vocalizations is still intact. And you often find the patients who have an expressive aphasia, because these lateral speech motor systems have been damaged. They've been damaged, they can't talk, but they still laugh, cry, often swear. These much more reactive emotional vocalizations that seem to be part of this older network. So in humans, you're seeing when people use their voices, often one system, somebody screaming because they think a spider jumped at them, or someone talking, those are two different networks. But of course, often you get the two interacting. And here we have an example of that happening. This is somebody talking on the Today programme. The pitch of her voices is shown down here.
Singarock's unpopular replacement has now been dismissed, with the Army's popular Chief of Staff, Jack Twat, taking over.
A 40-foot sperm whale, which was stranded in the Firth of Forth for more than 4 days, is now thought to be swimming towards open waters again. It freed itself late last night. Marine experts are hoping to establish this morning whether the whale is finally back at sea.
Good luck to the whale. 8:10 is the time. An investigation is underway at the meet.
So just before I go on, Robin, can I just confirm, could people hear that?
I could.
Excellent. So what you heard there was live radio. There was somebody coming down the line, and he's got to say what for certain parts of the UK is a funny name. So the word twat's quite a rude word in bits of the UK. Certainly where I grew up, it's very rude. And he just goes for it.
That wasn't what I heard, Sophie. I heard Whales.
I might go from the start then, okay? I'm just playing it from the beginning. See if the sound works for you this time. There are whales at the end.
Rock's unpopular replacement has now been dismissed, with the Army's popular Chief of Staff, Jack Twat, taking over.
A 40-foot sperm whale, which was stranded in the Firth of Forth for more than 4 days, is now thought to be swimming towards open waters again. It freed itself late last night. Marine experts are hoping to establish this morning whether the whale is finally back at sea.
Good luck to the whale. 8:10 is the time. An investigation is underway at the maze.
So hopefully you heard it all that time. The only point about the Jack Twat comment is that it is a bit funny. And if you listen very carefully, when they go back to the BBC studios, just before Charlotte Green, who's about to read the news headlines on this very serious news programme, somebody leans into her and they say one thing to her. They just whisper, Jack Twat, right at her. And they're doing that for one reason and one reason only. They are trying to make her laugh. She starts reading the news headlines and at first she's okay. And you can hear in the pitch of her voice, she's all right. And then the laughter starts to get her. And the first sign that you hear is that she starts to lose control of the pitch of her voice because different things are happening to her intercostal muscles and she's losing that fine subglottal control that's giving us our voices normally. She loses more and more control. The pitch of her voice goes stranger and stranger. Then it shoots right up. And by the end, she starts making squeaking noises after she stopped talking because now she's completely laughing. Now, I think this is an example both of how interesting it can be, the sort of latency of laughter, but also what you're actually hearing here are two different vocalization systems, one very old, one restricted to humans, more recent, which is duking it out. She is trying to talk and she's starting to laugh and you hear something happening. If you're not from the UK, you may not realize that she's starting to laugh, but you probably heard something happening in her voice. And then by the end, she's fully laughing. The laughter wins. The non-verbal, the older system does seem to be more powerful. It will generally win the battle in the end. Now, one of the other things, so if we just think about laughter in a bit more detail, laughter is an interesting behavior. It's a highly contagious behavior. This is a behavior, this is people being scanned, just listening to different kinds of emotional vocalizations in a study I did a few years ago. And what you see when people hear emotional vocalizations, and lots of these regions here in green, these orofacial mirror system regions, are recruited when you hear emotional vocalizations and when you move your face to make a sound. But interestingly, it's driven most of all by laughter. And we wondered if this was the kind of contagion of laughter, because frequently when people laugh, they are just laughing because they've heard other people laugh. It's part of this kind of suite of behaviorally contagious phenomena like yawning and scratching and blinking. But in fact, it's even more complicated than that. So in fact, even contagious laughter is a very social behavior. So you're much more likely to catch a laugh from someone you know than someone you don't know. And what we can see when we look at humans in laughter more generally is it remains an extremely social behavior. So Robert Provine found that when humans are 30 times more likely to laugh if there is somebody else with them than if they're on their own, which is quite extraordinary. I have to say the same is also true for voices. We don't tend to speak very much when we're on our own, but certainly laughter is highly potentiated by other people being there. So watching the same thing on your own, you're more likely to laugh much more if you watch exactly the same thing with somebody else. Now, this is interesting because we don't think that's what laughter is about. We think laughter is about humour. And again, Robert Provine showed this. So if you ask people what makes them laugh, they'll talk about jokes and comedy and humour, but if you actually look at people, it is mostly this a social behaviour. So we laugh most in conversation, and in fact, in the middle of human speech, which is where that is the dominant social mode even now for most human interactions around the world, and which really is a unique in nature. We drop into laughter a lot. That's where laughter lives. It lives in these social interactions. Everyone laughs more than they think they do. All of us underestimate how frequently we laugh. It's all socially modulated. So you laugh more when there's other people there. You laugh more if you know those people. You'll laugh more if you like those people. And in fact, within those social interactions, it's doing incredibly complex work. So we can laugh just because other people are laughing, this basic contagion. We'll also laugh to make and maintain social bonds, even transiently, even just as a smooth over a slightly difficult situation on a train, for example. In conversations, we laugh to show that we agree, we understand, we remember, we recognize. So we use laughter incredibly in a very, very nuanced way. And hardly ever in these interactions are we laughing at jokes. It's doing all this other kind of social work. And in fact, what Robert Profine found is that at any one time, the person who laughs the most in a conversation is the person who's talking. So we're using laughter in this very kind of a communicative way to get people to show that they understand, that they recognize, they agree with us. We will laugh to reframe things as play. It is a very important use of laughter. I'm not going to go into this in too much detail. I'm happy to take questions of it, but a very common use of laughter is to try and deal with stressful situations in a way that will make them okay, or make them acceptable, manageable. And I think one of the things that's quite interesting there is that's quite a common use of laughter in difficult situations. And if you look at how the evolution of how people have joked or not about the situation with coronavirus, that's been an example really of people trying to find different ways of using laughter to make the stressful situation less stressful. So what we have when we think about laughter is something that is happening primarily in social interactions. It is a social behavior. It's an emotion. It's a joyful emotion. It's a playful emotion, but it's an emotion that does not happen very much when we're on our own. It always has this social component. And the other thing, this is just two people who've come in for a study with us where we were actually looking at laughter in conversations. And these are two friends who've come in. And what you just notice, if you just watch these people talking, and they're being set up by the experimenter, is how often they just flash little bits of laughter at each other and little bits of smiles. So they're listening to Maria, they're getting ready to just do this study where they're going to be having a conversation, and they know they're being filmed. And as soon as she goes out of the room, they look at each other, and maybe this kind of reframing is play. But the thing to notice with this is how often the little flashes are coming and going really quickly. So little laugh there, she's gone, bigger laugh, that's gone. Now, if you think about it, they're gone. Both smiled, both laughed, stopped. That's very different from that laughter I played you of people on the radio trying to talk and that kind of duking out of the speech system and this older system. And we wondered, are there maybe different kinds of laughs? Are those sort of more communicative laughs, the laughs that are happening most of the time in conversations, are they different from that kind of really helpless, spontaneous laughter? There is a literature on this from the face. So if we look at smiles, if you look at these two pictures of Paul Ekman and think about which of these two smiling faces looks nicer, most people think the smile on the right looks nicer. And that's because that the picture on the left is a composite. So the mouths are the same. And what we've got here, the picture on the left has got eyes from what's called a neutral face, which just means a sort of a completely relaxed face. It's not particularly emotionally neutral, it's a little bit hostile. What it gives you is a smile on the left that looks a little bit less warm. And there is some data showing that when people smile to command, it looks more like the smile on the left, whereas when people smile spontaneously, it looks more like the smile on the right. So we asked this question about laughter. We got people in and we did whatever it took to make them laugh. Here's an example of somebody laughing. That's a spontaneous laugh. In fact, that's me laughing spontaneously. That's me laughing helplessly. And it's a completely, when that kind of laughter happens, it's an involuntary vocalization. I couldn't stop laughing by that point. And consistent with that, we make sounds when we're laughing in an involuntary way. We couldn't make under voluntary control. So I have a really high pitch laugh and I'd love to be able to sing that high. I can't. This is a more communicative laugh. We deliberately didn't want people going, ha, ha, ha. We wanted sort of warm, friendly, conversational laughter. And people are actually pretty good at spotting the difference there, because you could hear that laugh. It was lower in pitch. It was a bit shorter. But interestingly, certainly for our group of laughers, our kind of conversational laughter, our not spontaneous laughter, was quite often quite nasal kind of quality to it. So I think we're actually not even trying to fake it. We in this conversational laughter is perhaps its own thing. People are marking it as the thing that it is, and it lives in conversations where people very rarely stop and go, hang on, I don't think you're laughing helplessly. And the timing of it is very different. So when that spontaneous laughter happens, it's very hard to stop and it can be quite slow to start. Whereas the communicative laughter pops in and out really fast, can come and go, and it's doing its communicative job, we suspect. The brain is very good at telling the difference between this. We were hearing about theory of mind networks in the talk before mine, and we see those recruited when you hear laughter, but particularly When you hear laughter, that is this communicative laughter. These are the networks shown in pink. And in contrast, you get a lot more auditory cortex activation to the spontaneous laughter shown in blue. And this is not a study where people were being told to do anything. They're just being played sounds, and some of them were laughs, and some of them were authentic spontaneous laughs, and some are communicative. So even if it doesn't have anything to do with you, you are trying to work out what laughter means. And I think that's because it's so never neutral. Laughter is always meaningful. It is this very, very powerful communicative, social, emotional cue. And when you hear it, you're trying to work out what's going on. Interestingly, that contagion effect I showed you before was there for all the laughs. I sort of thought you might get more contagiousness for the spontaneous laughter, and it's not what we found. What we found instead was people who are better at telling the difference between spontaneous laughter and communicative laughter. When they were listening to the laughs, they were recruiting more of this kind of contagion, this priming response, than people who are less good. So it's not just contagion. If you are primed to join in when you hear laughter, it seems to help you understand what that laughter means. And that's probably meaningful because this is something we learn to do. These are people between the ages of sort of 3 and 80 rating laughter as this communicative or spontaneous. And what you find is children are not good at this, and you get better and better, but you don't really get very, very good at spotting the communicative laughter until you are into your late 30s. So this is probably something that is more of a social skill that you can only learn in interactions. And consistent with that, we find that this is just a study with boys who are at risk of psychopathy. They are high in callous and unemotional traits, and they also have conduct disorders. And what you find is that they don't find laughter as contagious, and they don't show the same brain response as boys of the same age who do not have these callous and unemotional traits. Now, you don't know the direction of causality here, but it certainly suggests that it would be worth looking a lot more at laughter, because if you do have issues or difficulties learning, or the opportunities to learn about this social emotion, it will have knock-on effects potentially, or be worth understanding in terms of interactions for your adult life. We've also found it's quite sticky. So we found adding laughter to any joke makes that joke seem funnier. And the more spontaneous the laughter, the funnier it makes the joke. And I think that's because, like I say, it's never neutral. And even if you hear a dreadful joke, if you hear some laughter, it seems to just make, well, perhaps the contagiousness, someone somewhere thought it was funny. Just to come back to this last point about what we're doing when we're speaking. So we tend to think about speech as being about where we're communicating information, and then we've got these non-verbal emotional sounds that we make. But I think there might be some real complexity there with laughter. So this is a study where Nadine Levan ran this, where we were asking people to tell, can you tell talkers apart? You've got two different people talking. Is it the same person or is it one person? Sorry, same person talking, or are they two different people? And we either had people listening to vowels, when you do that, people are pretty good. They can tell if there's one or more speakers there. And that's because there's lots of stuff that contributes to the fact that we all sound slightly different when we speak. If you did the same thing with spontaneous laughter, so you have two different people laughing, or the same person laughing, but the laughter is spontaneous, what you find is people are not good. People are not good at telling people apart, probably because spontaneous laughter can be so idiosyncratic and strange, even within one person. But interestingly, if the laughter is communicative, people get good again. So I think that suggests that some of this communicative laughter might have a lot more in common with speech than it does with non-verbal vocalizations, at least perhaps in terms of the cortical control, because you are being you, you're expressing yourself when you speak, and perhaps when you're laughing in this way. So I think there may be a bit more complexity here. We've got this older non-verbal emotional network associated with spontaneous expressions of emotion, including laughter. And we've got this more recent human adaptation of these lateral motor systems that underpin a lot of the complex stuff we do when we use our voice for speaking or beatboxing or impressions. But maybe laughter is an example of something that can vary from something that is spontaneous through to something that is recruiting much more this sort of communicative network, this voluntary network, this intentional network. And maybe that's something, it would be really, well, I say maybe, one of the things I would like to really be able to come back and tell you in a few years' time is exactly how this interaction works. When people laugh in conversation, is it always sort of bouncing back and forth between these two networks, or do we fully evolve a kind of learned a system for controlling a simulacrum of this spontaneous behaviour, which we can use communicatively. Thank you very much for your time, and I hope we have time for a few questions. Thank you.
Thank you very much, Sophie. Excellent as always. There are piles of questions coming in, so we're definitely not going to get through all of them. There's one here from Kingsley, which intrigues me, is how do you explain laughter associated with tears of joy?
Well, I suspect that there is something that I think Darwin was right, that there is something very joyful about laughter, and there's something truly sort of, you know, that you often do find yourself laughing, not exclusively, but at truly sort of beautiful moments. But there's something very interesting there about the tears, because humans are the only animals that produce emotional tears. So if you've got eyeballs, you need tears to lubricate them and to get rid of irritations. We also cry for emotional reasons. We'll cry with sadness. We'll cry when we're laughing. So for me, when I start laughing spontaneously, I immediately start crying, just jetting out tears. Some people cry when they're angry. And I think there's a whole other set of questions associated with that, like what actually is going on there, because we don't cry when we're disgusted. We don't cry when we're surprised. So there is something quite selective about when you do get these emotional tears. And I think there's something interesting, but a particular association with extremely unhappy feelings like crying with sadness, and extremely delighted feelings like crying with joy and humor. So there's a lot more to know about that. It's a very good question.
Okay, there's several questions here on cultural differences. I don't know if you can say anything about cultural differences, but also on the linkage between smiling and laughing. This is Ali Rigby here in particular, who says, I've read that ancient Romans didn't smile. That affect their capacity for laughter?
Well, there are cultural differences. So as far as we know, all human cultures laugh and recognize laughter. So it probably is, in Ekman's terms, a universal vocalization. But there are big cultural rules about when and where laughter is not appropriate. A good example would be the difference between the UK and Japan, where Japanese people laugh a lot, but there are more contexts in Japan than in the UK where laughter would be rude and people don't do it. So you don't laugh, particularly in the workplace in Japan, where it wouldn't be that unusual in the UK. But then other countries will have rules in different places. You know, Russian people tend to think that British people laugh all the time and look a bit stupid at work. So, and that's just, again, it's a threshold thing, like where is it is and is not acceptable. So every culture has a place where it's not appropriate, and that those thresholds are set in different places. The relationship with smiling is interesting, and again, the cultural roles there. So I've certainly read interesting stories that say smiling became very infra dig, certainly in the UK, when processed sugar became available for the Tudors and people's teeth started rotting. So you stopped showing your teeth when you smiled, and you did not laugh because, again, you would show your teeth. So that sort of became a rule about laughter. There's also probably a very interesting relationship between laughter and smiling in terms of smiles as a precursor to laughs. You know, like if you're talking to someone and for whatever reason, laughter might be appropriate, but you don't quite feel like it, like a smile is better than nothing, you know, so it has that kind of, you know, element.
Designed.
Yeah, or, you know, there's a book that's just come out about the Comedy Cellar in New York, and it's called something like, I don't want your applause, laugh or don't laugh. And that's the bottom line. I think probably there are also contexts when only laughter will do. And that would be perhaps more intimate, closer situations where the laughter has to be there or something's wrong.
Very good. Thank you very much. I think we really have to stop there. We've already eaten into the break time. So there will be a short, now 5-minute break, because we're going to start bang on 11 o'clock. And there's going to be a screen that'll go up and that'll come down when things start again in exactly 5 minutes from now. So we'll see you back after the advertisements, as they say. It's 11 o'clock. I guess we're live to go. Gillian, are you up and running? She's unmuted. Would you like to put your video on so we can, there you go, we got you. So the next speaker is Gillian Brown, who's the reader in psychology at the School of Psychology at the University of St Andrews. And she's going to talk about prenatal hormones and sex differences in human behavior.
Great, thank you. Let me find my PowerPoints.
Oops, we can't hear you.
Can you hear me now? Can you hear me now, Robin?
Gillian, we can't hear you. We've got your screen up and everything, but we can't
I can hear you.
hear you speak.
Can you hear me now, Robin?
No.
I can.
Okay, Sophie can hear me.
Yep.
Shall I just go for it?
We've lost the sound completely from you.
I think it might just be you, Robin.
Sophie, is your sound working?
Yes, I can hear Gillian.
Sophie?
Yes, I can hear her.
Your sound's working. Okay. And can you? Does that mean you can hear Gillian? Oh, okay. It's at my end. I'm going to. I'll disconnect and log back in. So off you go, Gillian. Go for it.
Great.
Ignore me.
Right. Let me find my PowerPoints again then. Lovely. Hopefully that's working. So, well, thank you to Robin when he can hear us, and to the other organisers for the invitation to speak today. I'm here in St Andrews, which is a small university town on the east coast of Scotland, about 50 miles north of Edinburgh. And we have sunshine here today, and I hope you do wherever you are, whether you're down in Oxford, around the rest of the UK, or around the world. And thank you for your time to come to my talk. I'm going to be talking about sex differences in behaviour, which is probably the most controversial topic that you could think of within psychology. And when we think about debates about sex differences in human behaviour in particular, we're often framing these in terms of dichotomies. So you might have heard of the arguments about whether differences between males and females might relate to biology or social environments, or you might have heard the term nature versus nurture. Or sometimes we hear about explanations based on evolution versus culture. And even when we use the term sex versus gender, we're producing another dichotomy, which suggests there's two ways of thinking about the behaviour that we see and why it might be there. My background is as a biologist studying natural sciences, zoology, specialising in animal behaviour and evolutionary approaches and mechanisms of behaviour. So you might think that I would favour one side of the argument versus the other. But in actual fact, what I'm wanting to argue is that what we need is a much more integrated evolutionary perspective that fully incorporates the roles of these multiple variables, including the social and cultural environments. So we go back to this term nature versus nurture, which has been around for at least 150 years now, originally through Galton. And we have this notion that genes and hormones have something to do with nature, and culture and socializations are the nurture part of how we develop. So what I would argue, and others have as well, is that we need to move away from this nature versus nurture dichotomy and think about how these multiple variables interact. Genes, hormones, culture, socialization, all within an evolutionary framework. So this is really my take-home message for the talk, and I'm going to try and talk you through some of my reasoning. And since my PhD days, I've been interested in hormones and how they might be involved as part of this larger picture. So what I'm going to do in this short talk is have a look at briefly how hormones might influence behavioural development, then show you some empirical research on rodents from other labs, but also my own lab. Just give a couple of examples about hormones are studied in human beings, and then have a think about which evolutionary approach to take, because there's multiple ways of thinking about evolution. In the bottom corner here, I've got a molecular structure of testosterone. It's a steroid hormone. Its structure is very closely related to other steroid hormones, such as progesterone and estrogens. And it's these hormones, sometimes known as sex hormones or reproductive hormones, that are particularly going to be the focus for us. So why might we think that steroid hormones have an impact on behaviour or on brain function? One of the reasons is that there's a lot of evidence for steroid hormone receptors being widely distributed in the brain. So this is just an illustration of estrogen receptors in the red triangles and progesterone receptors, the black circles, giving you an idea of where they might be found. And as you can see, they're in numerous different regions, including prefrontal cortex, hypothalamus, the amygdala, the hippocampus, the cerebellum, the brainstem, and androgen receptors have similar wide distribution as well. And cells that have steroid hormone receptors can be sensitive to those hormones. Hormones can have actions in cells that have the appropriate receptors. Hormones can also have impacts through non-receptor routes as well by directly impacting upon cellular and molecular processes. But this is good evidence that hormones can be affecting brain function. This picture in particular just shows how that relates to serotonergic pathways, but obviously these brain areas are actually involved in multiple different pathways. So there is some synthesis of steroid hormones locally within brain tissue, but it's also the case that hormones produced in the gonads at the other end of the body, so the testes and the ovaries, can enter the bloodstream and get circulated throughout the body, including up into the brain tissue, and they can cross that blood-brain barrier and diffuse locally within neural tissue. So these are hormones produced at one end of the body can potentially be having an impact on cellular functions, gene expression at the other end of the body in the brain. And research on behavioral neuroendocrinology, as the field is known, has shown that in adulthood, gonadal hormones that's produced by the gonads can have what's known as activational effects on behavior in a range of species. So as an adult, the circulating hormone levels in an individual at one moment in time might impact on the likelihood that that animal will engage in a particular behavior in response to a particular environmental or social cue. In addition, though, hormones are not just produced during adulthood, which is shown on the right-hand side here. You see they go up during puberty and remain high in adulthood. And the top panel here is showing an adult male testosterone levels as an illustration. The bottom panel is showing an example of estrogen levels in a human female. And you can see there's other times at which that individual is producing gonadal hormones. And so if we look at the top panel, before birth, there's a rise in circulating testosterone levels in human fetuses in the mid to late part of gestation. Levels then drop around the time of birth. There's a peak within the couple of hours of birth. And then there's this additional surge during around 3 to 4 months of age before levels drop again throughout the rest of childhood through to the pubertal rise. And there's a similar pattern in females in terms of the rise in estrogen levels before birth, during this early postnatal period, and then resuming again during puberty. And the left-hand part of these graphs are stretched out to show you that prenatal and early postnatal levels of hormones. And just to point out here that testosterone isn't a male hormone. Females also produce testosterone. It's just the average levels tend to be lower across all of these different stages of development. So females don't get exposed to this prenatal surge. And these hormones are being produced by the fetal testes and the ovaries themselves. And what research has shown is that these gonadal hormones can have organizational effects on brain development by influencing a broad range of neurodevelopmental processes, such as axon growth, synapse formation, apoptosis, so cell death, myelination. And the framework of talking about activational effects in adulthood and organizational effects during these early stages of life is a framework that's been within behavioral neuroscience, behavioral neuroendocrinology for the past 60 years. And this is really when the field took off in the late 1960s, early 1970s. And early research from this time showed that if you treat rats or mice or rhesus macaques with testosterone before birth, this can have long-term effects on behavioral development of females. And the reason females in particular is that the experimental paradigm is that pregnant rats or rhesus macaques in these early studies would be injected subcutaneously with testosterone, the mothers would. These hormones would then circulate through the maternal system, go through the placenta into the fetuses. And what you'd have in the female fetuses is you'd have raised levels of circulating testosterone, more similar to what you would have in the normal males. And then you'd have a look at those females after they're born through to adulthood to see whether there's any impact on behavior of being exposed to testosterone before birth. So is there any impact on behavior and brain function? And for example, the early rodent research showed there was a strong impact on later sexual behavior of these females that were exposed prenatally to testosterone. They showed an increase in masculine type mating patterns and a reduction in female type mating postures, for example. And over the 60-year period, a large volume of data is now accumulated to suggest that hormones have both these organizational and activational effects on behavior, particularly behavior related to male and female reproduction, for example. But I wanted to move a little bit further into development and think about adolescence as well, which has been an area that I've been interested in more recently. And during adolescence, there's a lot of evidence, including human beings, that the brain continues to develop during this period. In humans, it can continue through to people in their early 20s, as well as in their teenage years. And there's experimental evidence from non-human animals that the brain continues to be sensitive to gonadal hormones during this stage of life. So there's a potential for organizational effects of hormones to act during this sensitive period of development when there's a lot of brain reorganization going on. And we can study these kind of questions in non-human animals. So this is an illustration of testosterone levels in male Norway rats, laboratory rats. And this is from birth onwards. So again, they have this small surge postnatally at around 10 to 15 days of age. In most species, it's not completely understood what this postnatal surge is. It tends to be a reactivation of the hypothalamic pituitary gonadal axis, which then calms down again through juvenile stages. But just with humans, you then see the rises again in gonadal hormone levels during pubertal development that takes place during adolescence. So studying behavioral development in rodents is an interesting approach to take. And this is just some data from Professor Cheryl Sisk's lab at East Lansing at Michigan State University in the USA from their 2008 neuroscience paper. And this is looking at two areas of the hypothalamus. So we have the AVPV and the SDN, antroventral periventricular nucleus, and the sexually dimorphic nucleus. And these are areas which show differences between males and females in the average number of cells in these areas. And we're showing two types of staining here. And just to point to the SDN, so there's one side here, it's also on the other side here. And this has been studied since the early 1970s. And it's actually a difference which is observable just by looking at slices of rodent brain on a slide. So sections on a slide, you can see with the naked eye that there's a difference on average between male brains and female brains in the density of cells in this SDN. And what Cheryl Sisk's lab have done is look at when these differences emerge, and actually the difference in cell number is present there during this adolescent period. So this is postnatal day, these are quite young rats. And the males are shown here in the dark bars and the females in the light grey bars. And there's a higher density of cells, there's more cells in the SDN in males than females in these rats. And in comparison in the AVPV, there's a higher density of cells in female AVPV than males. And this difference is there already during the adolescent period. And what Cheryl's group did was to gonadectomize, so remove the testes or the ovaries of individuals before they went through pubertal development. So individuals either got a gonadectomy operation or they were sham individuals. And the left-hand part shows the AVPV, and this shows that gonadectomized females no longer show that higher density, that higher number of labeled cells in the AVPV. In contrast, in the SDN, if you castrate males before puberty, then they no longer show that higher level of cell number in the SDN, which strongly suggests that hormones produced by those gonads are having an impact on the brain in terms of maintaining cell number or increasing cell number or reducing cell death in those areas. And this is an interesting thing about hormones. They don't always show the same pattern in the same direction in different areas of the brain. So the hormones sometimes reduce cell death in some areas while enhancing cell death in others. So there's not one overall effect of hormones across these areas, but researchers are able to look at very specific regions and look at the impact of either having gonadal hormones during adolescence or not having them. And the Morishita et al. Paper, which came out earlier this year, shows that if you replace testosterone levels in these males, they used mice instead, if you replace the testosterone during this adolescent period, then you get a reinstatement of the normal pattern here, again showing that there's a difference related to hormones. So what my lab and I have done, we've mostly focused on looking at the behavioural effects of these hormone changes that occur during adolescence. And there's a lot of sex differences in behaviour that you can record in rats. We've looked at how they respond to novel environments, such as this one. This is a light dark box, so the animal starts in the dark section. And rodents tend to prefer to be in a dark area rather than out in the light, where there's more potential in the wild. There'll be more of a predator threat. So they spend most of the time in the dark area, but it's actually the females that will more likely to come out and explore in this open, exposed area. So the graph on the right shows that females are spending about 3 times as much time out in the light section that males are. We've also studied their play behavior, and these differences are actually ones that emerge during the adolescent period. So if you look at quite young rats, you don't find this difference, and it emerges during adolescence. We also looked at what happens if you carry out these castration type experimental designs, where some individuals will be castrated before puberty, some will be castrated after puberty, and you can have a long-term impact on these types of behavior patterns. So when we look at these adult animals, and these are all males, on the left-hand side, you can now see in the open bar that it's the ones that received prepubertal castration, so they weren't exposed to any gonadal hormones during adolescence, are showing a more female typical pattern in these novel environments. The left-hand side is a light dark box that I just showed you. The hashed panels on the graph will show those animals that are castrated after puberty. So in adult, when they're tested, neither have got circulating testosterone levels. The difference between them is whether they were or weren't exposed to those hormones during the adolescent phase. The right-hand graph shows a difference, piece of apparatus and elevated plasmase, which is very commonly used with rodent behavioural testing as well. And you get the same pattern of results there. Those who were pre-pubertally castrated are more likely to be going out and spending time on the open areas of that piece of apparatus. We also looked at social behaviour. So the males, again, that were either castrated before or after puberty, those that were exposed to testosterone during puberty, so the hash bars here, are spending more time in close proximity to an unknown female social partner, and the ones that were castrated before puberty, so haven't had testosterone exposure, appear to have a reduced motivation to engage with this unfamiliar female. And Cheryl Sisk's lab has also shown similar results, suggesting that exposure to testosterone during adolescence in male rats has long-term impacts on sexual motivation of these individuals. So again, we think that pubertal hormone levels are playing some role in terms of long-term behavior. We've also carried out some studies on females. There's no reason why organizational effects might just be limited to males. The ovaries, as we've shown, are also active at various stages of early development, and ovarian hormone levels again are starting to raise during adolescent periods. In contrast to the male data, so far we haven't found effects of altering hormone levels during adolescence in females. We've used another kind of design where you can give them a GnRH antagonist, which is gonadotrophin releasing hormone antagonist, which basically what it does is it blocks the receptors at the pituitary, binds to GnRH receptors at the pituitary, so it stops the normal GnRH pathway happening and just switches off gonadal hormone production. The pituitary needs to tell the gonads to produce hormones. If you provide an antagonist of the GnRH receptor, this system will reversibly shut down. So it's what we're doing is we're delaying the pubertal onset of hormone exposure by treatment with GnRH antagonists. These drugs are used in assisted reproductive technologies as well. And the graph just shows here, this is a different task. This is looking at how they respond to a startle noise in their environment. Control females on the right of the graph show relatively little response to these noises in their environment. They're much more interested in having a look around, trying to look for escape routes, see what they can find in their environment. The male adults are much more likely to freeze and show immobility in this task. And the AF group in the center are the females that received anti-treatment just during adolescence. So again, their hormone levels are reactivated in adulthood at the time of testing. They didn't get exposed to ovarian hormones during adolescence, but we didn't see any effect on this behavioral task or how they responded in novel environments or how they responded to social partners. So that's hopefully given you a feel for the kind of research that goes on in the field of behavioral neuroendocrinology to ask about hormone effects on development. I just wanted to put this slide up to show you that there are debates within the literature on the importance of including both male and female subjects in studies. And this is from a review paper in 2011. And the graph just shows a review of articles in various subdisciplines that were published in the year 2009. And in the blue, you can see the proportion of those papers, those published papers, where the subjects of those were only male animals, these preclinical studies. So these are mostly rodent studies. And you can see the third bar from the left is neuroscience papers. And around 50% of papers published in this year in neuroscience only used male subjects. Around a quarter of them either used both males and females or just females. And around a quarter actually failed to even mention what the sex of their subjects was in their studies. And there's been calls to much more consistently report sex and age of subjects in behavioral neuroscience literature. There are other fields where there's a better distribution, so behavior and zoology across the right, you're much more likely to see studies involving both male and female subjects. Immunology in particular, I think, is a field where there needs to be increased reporting of whether subjects are male and female. And there's actually an organization for the study of sex differences here, which is set up in the United States, which particularly focuses on the idea that studies of not just neuroscience, but all different physiological systems around the body would benefit from including male and female subjects. This is filtered through to clinical trials as well, and clinical studies, where there's much closer requirement now to include both males and females. If you went back 20, 30 years in clinical trials, it could be that you conduct clinical trials with only male subjects, and that would be allowable. But it's clear that if we're interested in drug development, then we need to know about how those drugs under development might impact differently on male versus female people who are taking them, because lots of systems in our body might function slightly differently across those two groups. I'm now going to move away from thinking about the animal research and think about applying these ideas to human beings. How can we think about development of behavior in our own species? And some researchers argue that actually hormones might perhaps have a minimal role in our species compared to the profound effects of socialization. So I think it would be very difficult to argue, and I certainly wouldn't, that socialization and culture have an enormous impact on what we expect in terms of gender roles and various cultures, in terms of what males and females are expected to do, what opportunities are available, how we raise children, for example. And Cordelia Fine's book in the right-hand corner, Delusions of Gender, I think gives a nice introductory and accessible summary of some of the research showing what dramatic effects aspects of our environment can have on how we behave and how we think about gender. I just wanted to bring out a quote from Cordelia's more recent book, Testosterone Rex, which I reviewed in Trends in Ecology and Evolution in 2017. And the quote here from the book is Fine arguing against the presumed role of the testes as the biological wellspring of the hormonal essence of masculinity, that steroid tsunami that destroys all hope of sex equality. And I've put this quote in to really point out the importance of how we ask these questions in human beings, because there are actual impacts on how we think about men and women, how we think about gender in actual everyday lives and in law and in education. So there are impacts on these ideas filtering through from the science to how societies think about sex equality or opportunities, or what our norms are about men and women. So I think a lot of these debates that you see in the literature are pointing out the fact that there are real world implications for how we think about the development of sex differences or similarities in human beings. In contrast to these researchers who really are arguing for the case for socialization being crucially important, probably the most largely influential factor in human behavior, others have suggested that actually early gonadal hormone exposure does actually play a role in human behavior. And I've chosen 3, again, accessible popular science books that have taken this approach. One that you might be familiar with is Simon Baron-Cohen's research on empathizing and autism. Here's a book from Joe Herbert, which talks about risk-taking, and a book by Melissa Hines in Cambridge, who talks about brain gender and how we can study individuals, particularly who are exposed to unusual hormone levels during development, how that might impact on play, for example. So again, we're coming back to this idea that within this debate, which can sometimes get quite acrimonious, there's this idea still of a nature versus nurture argument here. I just wanted to bring a couple of examples of the kind of research that's coming out from studies of human behavior. And one that's been very useful for researchers such as Melissa Hines, who are interested in early hormonal effects, is a disorder called congenital adrenal hyperplasia, or CAH. And individuals that have CAH have genetic mutations that impact upon enzymes within the adrenal axis, which is involved in cortisol production. And what happens is that mutations of these enzymes mean there's the shunting of hormone production from the normal cortisol pathway through to producing androgens again. These steroid hormones are very similar to each other. And if the enzyme pathway is involved in moving metabolizing one hormone into another on functioning, then you can get overproduction of a different hormone. And that's what happens in the adrenal glands of these individuals before birth. The adrenals start producing more androgens than they normally would. So females with CAH are exposed to higher average testosterone levels before birth than females without CAH. And the interest for researchers is looking at the behavior, cognition, development of gender identity in these individuals later on in life, and particularly looking at childhood play. There are several studies from several research groups now which suggest that the play behavior of females with CAH shows some similarities to play behavior of boys in terms of the amount of rough and tumble and engagement with those types of play activities. Obviously, there's some challenges in interpreting human data. These aren't experimental approaches, clearly. And there's also going to be a large amount of socialization effects in individuals with CAH. These are serious illnesses. These individuals have to have hormone treatments from birth onwards to correct these imbalances in the adrenal cortisol axis. And so they do have a different experience growing up, and they might feel different about themselves, and they might get treated differently by parents and others as a result. So we have to really acknowledge, particularly when we're studying humans, but also when we're studying other animals, that gonadal hormones won't act in the exclusion of socialization processes. These factors are likely to interact, and we need to start addressing them in multiple ways and take these interactions into account. I've put together this image, which I'm hopefully trying to show you how we might start thinking about development and how we might start thinking about hormones. So here we've got a behavioural trait on the notional x-axis here, and we've got development over an individual lifespan, starting in infancy, going through childhood into adulthood. And how we might think about hormones, particularly those before birth, is that maybe it just moves you along this top axis. So maybe you can slide along the top part of this, this diagram here in terms of where you are as an individual. It might be more likely that males tend to be on the left-hand part of this, this axis along the top, and maybe females tend to be on the right-hand part. But there'll be a lot of spread and variability. There's conditions where individuals are growing up with disorders of sexual development, which means they are somewhere along this, this span along the top. But then what I want to point out is that lots of things happen across an individual's lifespan that then influence how they go through development. And this can include crossing over of pathways, diverging, converging of pathways. And it might depend upon all sorts of things happening in the social environment, parenting, and we can try and impose some Waddington type landscapes on top of this diagram to explain how development occurs over time. And if we look at the child literature, there's a nice literature I just wanted to point you towards called dynamic systems theory, which really tries to focus on looking at how these behavior patterns might emerge over time. And there are various tools that developmental psychologists are using, such as state space grids, to look at how, for example, here, a child's emotional state and a parental emotional state might mean that it's more or less likely to move between one of these places on the grid rather than another place. And we could produce multiple grids for multiple individuals and start looking at whether there's patterns in terms of the likelihood of moving between these particular positions in these grids. Are some directions and movements more likely than others? We could think about hormones being involved in part of these movements across grid space, social environment, cultural environment as well. And just to point you to one study, researchers have started to use this to think about how hormones during adolescence might link with development of behavior or behavioral outcomes. And this Tunison et al. 2011 paper uses this type of approach to look at the likelihood of expressing symptoms of depression during adolescence. They had longitudinal data is quite useful in these settings, for example. And you can look at how the stage of development, so early, relatively early stage of pubertal development compared to your peers, can increase the likelihood of showing symptoms of depression, but it also relates to things like social popularity among peers. So it's not just to do with the state of which you are through your pubertal development. It can also be influenced by socialization or social factors such as popularity. So child development tools like this give us an opportunity to start producing slightly more complex patterns of how behavior can emerge. I just wanted to take one moment before I finish on thinking back about evolution to point to the digit ratio literature, which you might have come across, and you might all be looking at your hands now in front of you, in front of the screens. But there's a very small difference in some data sets in terms of the relative lengths of the second finger and the fourth finger. So in females, it tends to be that these two fingers are quite similar in length. It tends to be slightly more likely, but these differences are very marginal, that males will have a shorter index, so second finger compared to the fourth finger. But all I want to say about this literature is that people have suggested that this digit length ratio relates to prenatal hormone differences. And there's a rationale for thinking in terms of this androgen receptors in the digits as they develop very early in life. But there's really no good evidence that the amount of exposure to testosterone relates to digit ratio in later life. So I would be very wary about this literature. There really isn't a strong link showing a direct relationship between, or even a probabilistic relationship between digit ratio length and prenatal hormones. So I would caution you against this, the digit ratio literature. So I just want to finish up thinking about evolutionary approaches, and I will point you, if you are more interested in this area, to a book that I co-authored with my colleague in St Andrews, Kevin Leyland, called Sense and Nonsense, Evolutionary Perspectives on Human Behaviour. This is the second edition from 2011. So in this book, what we do is we critically evaluate the various evolutionary perspectives that people can take on human behaviour. I don't have time to go into it today. I just wanted to raise this because I want to link it back to this question about hormones. So you might have heard of the field of evolutionary psychology. This is just one of the approaches that we look at in the book. And evolutionary psychology takes a very essentialistic view of sex differences. It's this idea there's an essence of male or female that might be due to genes, might be due to early hormone effects, and that it gives rise to evolved psychological mechanisms that differ on average between males and females as a result of selection on our ancestral human populations. Okay, so this is, you might have come across this a sort of popular science view of evolutionary psychology. But I've been quite critical of this approach, as has others in the field. I've written several papers showing how this is quite a limited perspective on how we might think about evolution of human behaviour. And I just want to briefly talk you through that for a moment and then wrap up with how that impacts how we think about hormones. So I think we need to go back to this idea that human beings have these two inheritance systems. So on the left here, from time 1 to time 2, we've got the genes, the genetic information, the genotypes of a population being passed on from one generation, and then what you find in the following generation, given births and deaths and reproduction. And on the right-hand side, you've also got the behaviour of that population. And this could be an animal species, this could be a human population. You've got behavioural patterns at time 1, and you've got behavioural patterns at time 2, part of which will relate to socially transmitted information, what we learn culturally. If we think about evolutionary psychology, the focus there is really on these dark arrows going across between these two systems, from genes to behavioural phenotypes. So evolutionary psychology is very much taking a traditional view of evolution on genes, how that might relate to these external environments, and doesn't really put much effect on socially transmitted information. And I'm just going to skip through cultural evolution, which will focus more strongly on the transmission of cultural traits, to give you this slightly more complex pattern of gene culture coevolution. And what I particularly wanted to point out with this bidirectional relationship between genes and gene flow in populations and behavior and the behavioral phenotypes. And we have to take into account that behavioral phenotypes and behavior can impact on the processes involved in genetic evolution, that there are these bidirectional interactions. And the reason why this is interesting is that this plasticity of behavior, the variability we see in behavior, is partly underpinned by these hormonal processes. So hormones and what they're doing across lifetime periods of individuals and evolutionary time periods can impact on evolutionary processes. I'm just going to leave you with this, directing you towards the Extended Evolutionary Synthesis website, which would give you more information if you're interested in this approach. And I'm going to wrap it up there with just some thanks to collaborators and funders, and thank you for your time, and I'm willing to take questions. Thank you.
Thank you very much, Gillian. We're very tight for time here, so I think we're just going to pick one question at random. And this comes from S. Tumbeva. If I can find it again, because it's flipping up with questions flooding in. Can epigenetics explain why siblings turn out to be so different?
Okay, I didn't mention epigenetics, so that definitely comes into the latter part of it, though, thinking about developmental plasticity and extended evolutionary ideas about how transmission of behaviour might occur across generations. So we can think about maternal effects impacting on the behaviour and development of offspring. I think in terms of role of hormones in epigenetics, it's important to know that hormones can influence epigenetic processes. So absolutely, I think hormones in epigenetics could give rise to some of this variability we see between sexes, but also definitely between individuals within sexes as well.
Which prompts me actually just very quickly, because I'm taking time from myself, just to throw in another kind of supplementary question, really. And this is what struck me when you were talking, is having hormones having such an important role in developmental processes seems to expose the system to extraordinary environmental vagaries. If you think of sort of most normal developmental processes trying to achieve some vaguely consistent pattern, we all look sort of the same. You know, the hormones seem to sort of throw spanners in the works, and that makes me think actually must be a very good evolutionary reason then in that case for that. I just wonder if you have any suggestions on that front.
Absolutely, it's a really great point. And actually, you know, the reason for having endocrine systems at all is to allow those interactions between what's going on inside the body and what's going on externally. So it's actually, they're actually there to engage in some of these interactions and to do this sort of fine tuning between these different systems. As you say, if the system's too flexible, that might be selected against, but actually hormones are there to allow that flexibility. And I think you're right, we shouldn't think about hormones resulting in a hard wiring or pre-programming. Those are both terms that I have real issues with. Hormones are much more interesting than that, because that give that potential flexibility across individual lifespans and evolutionary time periods as well.
Okay, thank you very much, Gillian. And we better move on to the last of our talks, which is me. If I can manage to call my screen up. So I'm going to talk about social bonding, really, and some of the neurobiology that underpins that, some of the work we've done on this topic. So the issue really goes back, I suppose, to the fact that we have, relatively speaking, but at least by primate standards, very large social networks, personal social networks. Ours typically runs out to about 150 people or thereabouts. This is really 3 times bigger than the largest social networks or social groups you find in other monkeys and apes, and many have much smaller ones. This pattern of layers that you see here it turns out to be extremely consistent, and the layers and the numbers that are included in the layers really reflect the quality of friendships and indeed in many ways the levels of prosociality and altruism that people will exhibit towards each other. They also reflect the time investment that you put in your relationships. So that inner core of 5 close friends, or I sometimes call the shoulders to cry on friends, which are typically half family, extended family, and half true friends, they receive between them 40% of your total time investment in social time. And the people that make up the 15 layer, which includes the inner core of 5 there, so the extra 10 people get another 20%. So that those first 2 or 3 layers there, out to best friends, absorb something like 60% of your total time investment. They're hugely costly to you, and there are very good reasons why you need really to invest so much time in them, which I'll come to. But just to show you how consistent these are, here are the same data from different sort of media, Facebook, Twitter accounts. So these are the Twitterati, the followers of a Twitter account, talking to each other in directed posts. Cell phone calling patterns, so the frequency with which people call the people on their phone address books, as it were, as well as these face-to-face layers that I've shown you on the main screen. You can see just how consistent these patterns are across these different media. The effects are extremely robust. So the question is that I'm going to focus on really is how do we maintain these relationships. How does it actually work inside the brain, what's going on? And the model we've developed for non-human primates is what's sometimes called a two-process model. Let's say there are two mechanisms involved which are running in parallel to each other, both of which play an equally important role. One is a very deep neurobiological process that's emotionally very intense, tends to involve what the philosophers used to call raw feels. Let's say you have these sort of feelings about things, but you often can't put them into words. And this seems to be associated in primates in particular with social grooming, of which there are several examples illustrated here. So monkeys and apes devote an inordinate amount of time to this activity. Some of the more intensely social species will spend a fifth of their entire day doing this otherwise useless activity. Well, obviously it helps keep the fur clean, but they're doing it a lot more in those cases than they just need to keep the fur clean. But this seems to then sort of provide a kind of pharmacological platform, if you like, off which a cognitive component is built, which is associated with the development of relationships of trust and obligation, reciprocity, and in here, mentalizing, which several of the talks have already mentioned, becomes very important. And this seems to be associated with the size of neural circuitry in the brain, very specific neural circuitry in the brain. And I'm going to talk about both of these primarily as they then apply to humans. So I'll do them kind of in reverse order because I'm going to say a lot less about the cognitive side. Jerome in his first talk today mentioned mentalizing or theory of mind. Although what he focused on was what's technically formal theory of mind, which is this sort of middle row here, where Jill's thinking about what Jack is thinking about. So Jill's thinking about what the contents of Jack's mind. But in fact, in humans, this, actually, I should say that formal theory of mind in this sort of central block here, thinking about somebody else's mental states, is something that is pretty much unique to humans, but great apes can have a jolly good go at this. They're pretty much on the verge of passing these kind of tests. All other animals, including all the other monkeys, sit down here or scattered in between these two things. But humans can go further than this. They can embed successive series of mental states, and the sort of limit seems to be at about level 5. So you can understand 4 other people's mind states as they're thinking about each other. So it's just sort of really setting the scene here, because what we've done, and a number of other people have now done, is look at the neuroimaging correlates of these mentalizing state capacities, and particularly in relation to the number of people in people's social networks. And that's been, social networks have been measured in as many different ways as you can think of. Some studies have used the number of friends on Facebook pages, some have used the number of friends that you have in your total social network, that essentially averages out about 150 across the population. Other studies have looked at the inner core layers, and you get pretty much the same story with all of them. Jerome has already talked us through that. It's this neural circuitry that involves the prefrontal cortex up here, various units in the prefrontal cortex, with the temporoposterior junction here, and the, where are we, down here, the temporal lobe, and then associated linkages into the limbic system, the amygdala in particular. And that generally seems to go these days under the name of the default mode network. And that seems to be absolutely crucial to managing both mentalizing states and the size of your social circle. So there's about a dozen neuroimaging studies now that have shown correlations between the sizes of those components and the size of your social network, and about 5, perhaps 4 or 5 of those studies have looked at mentalizing capacities. So the more mind states you can handle, the bigger these areas are, these units are. And in one of those studies which we ran, we did a path analysis which tried to unpick the causal sequence apart, and the causal sequence seems to go from the size of these default mode network units to mentalizing capacity to the number of friends you can have. Okay, interestingly, Jerome mentioned his study of macaques, showing that there's a correlation between the size of housing groups that their macaques are housed in, and the size of what is essentially the same network. And there's another study coming out, also this one from Marseille, on baboons, another old world monkey, showing very, very similar results over long-term period as well. We've tried to unpick this a bit with PET scanning to look at the signature of attachment style in this context. So this is just using a standard psychologist attachment style. This is one of the dimensions, the avoidance dimensions. So essentially you are warm and cozy up here, and cool and offhandish down here. And the density in particular, and I'll come back to say more about these later, of the endorphin receptors in these in core areas, and particularly up here in the orbital frontal cortex here, but also some interesting areas like the thalamus, are correlated very strongly with how you score on these kind of psychometric measures, as it were. So people who are at the warm and cozy end of attachment and social style tend to have very high density of endorphin receptors in these core areas associated with the default mode network. So that's all I'm going to say about the brain units in this particular. I want to really focus most of the time I've got on the neurohormone systems involved, and the question of why time itself is so important. So this is the psychopharmacological platform, as it were, that seems to support social relationships in primates in general. And just to emphasize how important the time investment is, this is data from one of our studies where we followed people's social relationships and their frequencies which they contacted the members of their social circles over an 18-month period. These are actually people in their last year at school here, just before A-level, taking A-level high school exams before going to university. They go to university at about the middle point here in the summer, or sorry, end of the summer. And so this is the whole of their first year at university. And we're measuring their sense of emotional closeness to all the members of their social network, and they're split into their extended family group, their kin, and their true friends. And I just want to emphasize here this dramatic fall off in the sense of emotional closeness to friends after you've moved away from them and can't see them. This is a very widely documented effect in social networks analysis, but it's very, very striking. I would point out, for the benefit of everybody, though, how stable kinship relationships actually are by comparison. There's something very different between your relationships with your family and your relationships with your friends. But friends are very fragile, friendships are very fragile. You have to invest in them very heavily. And the burden of that investment really comes from this social grooming activity that we find in primates. Now, one of the original core experiments that was done to show how important endorphins, or in particular, are as underpinning this, it was done by Barry Cavern in Cambridge, in which they injected very low doses of morphine, subclinical doses of morphine into monkeys, and looked at the pattern of demand for grooming partners that the animal showed, and you can see this dramatic drop in their interest in grooming after they've received a morphine dose. And they did the converse experiment, which was give them an endorphin blocker, naltrexone, which locks onto the endorphin receptor sites in the brains and blocks the receptors to the activation by the morphine. And you can see grooming demand is just increasing dramatically. They just can't get enough of it. No matter how much they get, they want more. Morphine is a natural sister chemical of the endorphins. Endorphins in the brain, the name derives from endogenous morphine, meaning the brain's own morphine. So chemically, they're very, very similar, except that we don't get addicted to endorphins in the way we get addicted to morphine. But the point here is this system really seems to underpin primate social friendships, if you like, and seems to be important in building relationships of trust. We can show exactly the same effects in humans. So with our imaging collaborators in Finland, Lauri Numenmaa and his group, we ran a whole series of studies on adult humans in PET scanners. We did it on males because we figured as the less social sex, if we could show an effect at all in males, we could show it in practically anything. That was making it a fairly tough test. So we had males in the scanners, we had their partners, giving them very light, slow stroking. And here's the brain firing up, the endorphin receptors in the brain just firing up in response to just this activity. So it's the contrast is between no activity and stroking. So the brain is absolutely everywhere, but particularly up in this prefrontal cortex, up around the temporal junction and in the temporal cortex itself elsewhere, but not at the back here in the occipital cortex, of course, which is just primarily vision. So there's nothing of any interest socially, if you like, at that point. Vision only becomes interesting as the signals get pushed up towards the front of the brain and meaning gets attached to them. The mechanism that underpins this is only relatively recently been discovered, and that's the afferent C tactile neuron system, which is rather unusual by comparison with most or all the other peripheral nerve systems, in that most of the other peripheral nerves are fast, they're myelinated, they respond very rapidly to peripheral sensations, such as something creating pain, like a pinprick. They also have a return loop, which comes back down to the motor neuron, to the muscles in the same part of the body here, the hand, causing you to withdraw your hand from the circumstance that's causing the pain. The CT neurons are completely different. They are unmyelinated, which makes them extremely slow. They have no return loop at all. They go only one way, and they go up into the insular cortex up here, which is one of the main areas triggering endorphin output. And they respond to one stimulus and one stimulus only, and that is light, slow stroking at exactly 2.5 centimeters per second. So if you stroke somebody's hand at 2.5 to 3 centimeters per second, you'll get an endorphin rush in the brain. If you do it at 30 centimeters per second, you'll get no response at all. Incidentally, it's recently been shown that there's a very high density of these receptors in the inner ear. And we've just shown that if you shake your head a lot in listening to music, for example, it gives you a very big kick in endorphin output in the brain, at least as measured by differences in pain levels. One of the things that kind of bothered us a lot when we started to think about the neuropeptides underpinnings of social behavior in primates and humans was the fact that the literature seemed very messy, and that there was a lot of scattered, it was a very large literature, but it was very kind of scattered in its focus. You had a lot of people looking at different neurohormones. Gillian mentioned testosterone earlier in her talk. And no one ever controlled for production of any of the others. There's been a lot of focus and excitement and media hype on the role of oxytocin, the love hormone as it's come to be known in the popular literature, and using pain threshold changes in effect as one of the measures for its production in the system, completely ignoring the fact that the beta endorphin system does exactly the same thing and is triggered by exactly the same factors, behaviors. And also the other thing that bothered us was the fact that almost all the work had been done only on dyadic relationships. And in primates in particular, live in a much more complex world than simple dyadic relationships, you and your romantic partner, or you and your offspring, or you and your best friend, which is what most of the literature focuses on. Those dyadic relationships are embedded in very large, wide social networks, as I implied at the beginning. But also at the back of that, and it's much back to Gillian's territory perhaps, there is the social dispositions that we have as individuals, and that relates back to the attachment style story that I mentioned earlier. So to try and sort this out, what we did was to run a very large genetic screening study. We had about just over 1,000 subjects all told. They provided us all with SPIP samples, and we had these assayed for 35 SNPs for this, what seemed to us to be the 6 main social neurohormones. So these are them here. And they also then at the time filled in a whole bunch of questionnaires relating to measuring their social predisposition, if you like, how warm or cold they were. That was one of them. The quality of their dyadic relationships, how attached they were with the person, how reliable, if you like, their relationships were, how stable their relationships were, and then the measures of their wider social network and the size of their friendship circles and so on. And here's the pattern we get out. This is a correlational studies looking at which alleles of these various, the receptors for these various neurohormones correlate with these 3 levels of behavior, if you like. And each of them, bear in mind, each of these levels are based on 2 or more different measures averaged together appropriately. And you can see here that beta endorphin really very strongly affects your predisposition as this testosterone, but in terms of dyadic relationships and a lesser extent wider networks, beta endorphin is really one of the one that has the key effect here. Oxytocin comes out strongly, but it's only works in terms of dyadic relationships, and actually that correlation goes down dramatically if you factor in control for beta endorphin alleles at the same time. And then dopamine is the other strong effect, and clearly has a strong effect on the size of your wider network as opposed to your kind of predispositions. What's interesting about these two is beta endorphins and dopamine work in tandem. If you hit one, you'll hit the other. And if you suppress one, you'll suppress the other, which isn't always true for any of these other ones. So these two seem to be very important in regulating your social behaviors, and they work, if you like, at complementary levels. With the other 4 neurohormones that we looked at here having either very small or minor and very specific relationships. So now to the role of time. The burden of this goes back simply to the fact that in primates, grooming time increases with group size. In general, linearly, although it flattens off, as you can see here, at around about 20% of total data. I'm so sorry, the label's missing off here. That's the percentage of the total day devoted to social grooming. So we're interested really in how much time we should devote to social interaction if we were bonding our groups the same way that primates do, namely through social grooming, with our groups of about 150. And if you just take that up to the linear relationship that you have for these data, then we should spend about 45% of our day in social interaction. Well, that's physically not possible. And in any case, if you look at the amount of times people devote to social interactions, and we have found a sample of 7 different data sets from different societies from all over the world in the literature, the average was exactly 20%. So it looks like we're spending exactly the same amount of maximum time that primates devote to social interaction, bearing in mind that the other 80% is what they need for foraging and for resting and all the other requirements of keeping body and soul together. That's what's setting this upper limit. That we have the same upper limit, but somehow we use that time more efficiently to effectively raise the equivalent amount of time up to that required for 45%. So the question is, how do we do it? Well, one of the essential problems here is that grooming in monkeys and in humans is very much a one-on-one activity. You cannot groom with several individuals simultaneously. Very, very occasionally happens. It doesn't last very long. And I invite you to try cuddling with 2 people in the back row of the cinema, and I'll bet you a bottle of good wine that one of them will walk out within about 5 or 10 minutes because you haven't been paying them enough attention. It's a very, very one-on-one focused activity, this primate form of social grooming that we still do in our closer relationships, of course. So here's the problem. This is essentially the projected amount of social grooming time up here that we would expect for all our fossil ancestors. So each of these data points is a population of fossils, including the modern humans up here, plotted against time. So this is on the basis of their brain size, extrapolating through from brain size to social group size to grooming time requirements from the previous graph. This is what you'd expect them to have. Here we are, modern humans sitting very nicely at about 45% of time devoted to social grooming, whereas the early species, Australopithecines, are sitting very comfortably around this 20% upper limit. And what we think has happened is that over the course of evolutionary time, we've introduced these various activities as part of our social toolkit to broaden out effectively the number of individuals you can groom with at the same time, because they don't involve physical contact directly, and therefore you can, if you like, groom with several individuals simultaneously, or virtually groom with several individuals simultaneously. The earliest of these is definitely laughter. Sophie spoke a lot about that. It's a very visceral, contagious effect that we share with great apes. Later, we think, came in singing and dancing, singing without words here. And then once you have language, religion, storytelling, and indeed feasting. And they probably came in at these critical time points here where there appear to be dramatic changes in the pattern of the fossils. Okay, so just to show you that these things really do work in this context, and I just emphasize here that what's important about all of these activities here, including storytelling here, the rituals of religion, all of them trigger the endorphin system. We've done this mostly through looking at pain threshold changes, pain threshold rise as a result of activation of the endorphin system in all these activities. I'll show you some of these data plus some neuroimaging corroborating data. So the earliest one we did was really laughter. We just show people a clip of stand-up comedy for 15 minutes or so. To measure their endorphin output, we use the pain threshold changes. So we give them a pain threshold change in some way. How long can you hold one of these blood pressure monitors blown up to full capacity on your arm? They watch the video and then you retest for pain. If endorphins have been pumped out, pain threshold should change across this. So pain thresholds here should be higher than here. If there's no endorphins, there should be no change or maybe a negative change. We always use something boring like golfing instruction videos as the control against which to compare this. And then this is data from 5 of our experiments, all very similar. Here's the change for comedy, people watching comedy videos. It's all positive, so it's above 0. Negative changes in pretty much all the people watching boring control videos. And we've done it again with our collaborators in Finland. PET scanning to look at the endorphin activation in the brain. You can see the brain is absolutely going wild, particularly up in these cortical regions, especially the prefrontal cortex again, but also a few other areas in the brainstem. Again, nothing at all absolutely dead in the occipital lobe, the visual system, and even the cerebellum. But essentially the brain these are the receptors, if you like, absorbing endorphins like fury in response to laughter. We've done very similar studies with singing and dancing. These are data from dancing. One of the studies we did was in Brazil. We measured pain threshold changes in response to synchronized dancing or unsynchronized dancing and low exertion, high exertion. So this is full body dancing, very simple dance moves. And this is just sitting on a chair doing the same arm movements, but not actually much physical movement. And here's 0 change in pain threshold. And you can see this dramatic increase in pain threshold, especially when you're involved in high synchrony, high energy dancing, and in synchrony with other people. So these were doing it in groups of 3 or 4 people together. And we, just to show that this has its consequences for bonding, we also asked them to measure their sense of bonding to the other members of the group they were dancing with. So it's not to their friends, it's the people they're dancing with. And you can see the same, a very strong correlation here between pain threshold changes reflecting endorphin activation and their sense of bonding or belonging to the other individuals. Now, what's interesting about this is when we asked them to rate their bonding by the same index to their friends who weren't there on the day, it was completely flat. This is something very specific to the people you're actually doing these activities with. And we just to add to that, we subsequently then ran an endorphin antagonism study using naltrexone to block the receptor sites in the brain, just to show that it really was endorphins that are active, the active ingredient here. And we've done the same for storytelling, for religious rituals, attendance at religious churches and the like, as well as singing, and we get the same story across all of them. Somehow the these other activities that we've lit on, which just happen to activate the endorphin system, and I stress that with the exception of laughter, the others are kind of cultural things we've lit onto and grabbed hold of, if you like, in the course of recent historical evolution, as opposed to deep evolution, to increase the size of our social groups and the friendship groups. So just to sum up, social bonding involves both a neurocognitive and a pharmacological mechanism acting in tandem. It's a classic 2-process mechanism. Essentially, we use the same underpinning mechanisms that primates use, particularly for close relationships, and that we've found ways of triggering the endorphin system to supplement these ancestral grooming mechanisms, so we can groom with more people at once and allow larger social groups to occur. I think that's where I end, and so let's close all that down. Right, that can go. Where are we? Hopefully, okay. So we've got a couple of minutes left. I'll see what. All right. Somebody's asked if there's a role for anandamide, and the answer is not that I know of. I've no idea. But if you can actually tell me what its ingredients are, I might be able to say. Rituja here asks, is it in fact true that the distance relationships have a higher chance of breaking apart than the people who live together? And the answer is yes. It takes time. What seems to happen more is that relationships just fade quietly away if you don't engage in social activities with people that you consider to be friends. And I might add that the kind of digital media like Zoom work reasonably well, but they seem, so far as we can tell, only to be sticking plasters. In the end, it's face-to-face interactions that really work to keep relationships going. So you have to be able to literally go and hold on to somebody to once in a while to keep that relationship going. Otherwise, no digital media will stop them breaking down. Does laughter on phone calls or virtual environments also increase pain threshold? That we've never looked at, and I don't think anybody has actually looked at. I would guess so. What we do know from our research, though, is that people find phone calls and Skype and even digital text-based media like text and so on more satisfying and enjoyable if there's laughter, whether that laughter is real laughter or virtual laughter like an emoticon or something like that. So I suspect it's very likely there's a good project there for somebody to do. Humans and other primates use hands a lot, but the grooming behavior is related mostly to the C-tactile fibers, somebody points out, and the glabrous skin has no C-tactile neurons. How do you explain that? That was certainly our belief, but there has recently been a study suggesting that in fact the glabrous skin, the hairless skin like the palms of your hands, actually does have C-tactile fibers in it. I think mostly these C-tactile fibers on the hairy skin are attached to the base of the hair follicles, but what actually causes the effect is the physical movement of the skin over the receptors, as it were, for the fibers, rather than anything per se to do with the hair being moved, although that's going to have an effect. That being the case, then your hand actually, the surface of your hand, which has no hair on it, may still be moved. And that's exactly what seems to happen with this very, very high density of C-tactile type neurons in the inner ear. That obviously there's no hairs to be moved there. It's just the effect of the auditory system moving the neurons as it receives signals from outside. Would you expect differences in genetic predisposition across different genetic ancestries? The answer is yes. I guess so. We know that's the case for the endorphin system, at least between Asians, East Asians, and Europeans, in that East Asians seem to have a much greater sensitivity to endorphins than Europeans do. But that, I don't think anybody's really looked at that in any detail. And let's see, one last question, because time has really run out. Does virtual bonding make up for the distance in case of long distance relationships? Well, actually, I mentioned that the answer is yes, up to a point, but it doesn't seem to have sticking power. It works like a plaster to keep the thing ticking over, but it doesn't stop relationships in the end dying very quietly, which is what seemed to happen. So I better stop there. Thank you very much for all your questions on all the talks, and thank you very much to all the speakers. Before we finish, Jasper Hunt of the Cortex Club would just like to say something. So Jasper, if you would like to unmute yourself and show us your, there we go. Take it away.
Thank you so much, and thank you for the wonderful presentation. Give me just 1 second to get my screen share on. Here we are. All right, so I'm here to talk about the Cortex Club. You heard from my co-president, Tara, yesterday, and it'll be just about 5 minutes talking about what we do and encouraging you to sign up for our mailing list and attend our events. So the Cortex Club is Oxford's student postdoc neuroscience society, and although our committee is formed of students at Oxford, our events are free and open to the public. And especially now that we've gone virtual due to the COVID-19 pandemic, we would encourage neuroscientists from all over the world to come and view our programming and see what we have to offer. And just one quick note about what we do, because this is a frequent misconception. We talk about all kinds of neuroscience and not just the cortex, but we're called the Cortex Club, but we're interested in all parts of the nervous system. And so we have 3 kinds of events. We're at the intersection of communicating scientific facts and discoveries, like through seminars, providing career development opportunities, and talking about scientific policy, culture, and advocacy. And so I'm going to mention one upcoming event that we've got in each of these 3 areas over the upcoming term. So on the 15th of October, we're hosting a seminar by Bernardo Sabatini at 2 p.m. London time. And we're really excited for this event, and we would highly encourage everyone to come. Again, it's going to be free to everyone. Just sign up to our mailing list the day before and you're able to attend, and we'll distribute the Zoom and YouTube links to attend the talk. Something that we have in terms of career development is our Emerging Scientists Seminar Series. We identified that there's kind of a gap in presentation opportunities for people who are near the end of their PhD, but maybe starting their postdoc. And so this is a seminar series that's open to people in the last couple years of their PhD or the first couple years of their postdoc. And our first event in the Emerging Scientists Seminar Series this term will be on the 20th of October. Christiana Björkli of the Norwegian University of Science and Technology and Siddharth Ramanan from Cambridge University are going to be telling us about their fascinating research on Alzheimer's disease. And so we would love if you were able to attend that and come with good questions. And our call for proposals is open, and we accept applications on a rolling basis. So on our website, you can find where to submit an abstract if you would be interested in giving one of these talks for us. And then lastly, on the 10th of November, we're going to be having a panel on reproducibility in neuroscience, because we want to stay engaged with these questions of scientific policy, culture, advocacy, and meta-science, asking how can we do the best science? And as scientists, how can we study the ways that we do science and do better science? And for that panel, we'll have Dorothy Bishop, Verena Heiss, Russ Pauldreck, and Guillaume Rousselet. And so I'm really excited about this event, and we would love to have you there. You can sign up for our mailing list to join our events. Please sign up with an institutional email address, so something at .edu or just your university email address or company email address. for all personal email addresses, we have to go in and manually verify that the person is legit to keep our events secure from potential spammers and Zoom bombers, and that makes a lot more work for us. So we would really appreciate if you could sign up with an institutional email address. And the steps on how to join us are on our website, which is just cortexclub.com. You can follow us on social media to get notifications about our events and see what kinds of things we're planning and what kinds of how we're engaging with the neuroscience community. And you can check out our podcast, CortexCast, which in which we interview neuroscientists about their work, sometimes about their personal life a little bit. And with that, I want to say, have a good break, and I'm looking forward to more great programming with the Autumn School in Neuroscience.
Thank you very much, Jasper. I guess that's the end of the morning session, and you can all grab a sandwich or have some lunch before everything reconvenes again at 1 o'clock. Do you want to add anything, Masood?
No, thank you for a wonderful session, Robin, and all your speakers. And as you say, we'll be back at 1 o'clock with our last session, which is on neuroanatomy, MRI to microscopy. So we'll see you then. Thanks a lot, Robin.
Okay. Bye, everybody.